This month we are doing these genome design, genome analytics short course sessions. There'll be 4 of these. This is the first one on constraint-based modeling. We're kind of in a strange spot where some of the people here are very familiar with these things and some people less so. So I'm going to be describing these things as if you have no idea what we're doing here. So please forgive me for being redundant if you're very familiar with all these programs and technologies and everything. But at the Center for Biosustainability in Denmark, the genome design and genome analytics group are part of a design, build, test, learn loop for building microbial cell factories. And specifically, our job at UCSD is to build computational tools to accelerate a strain design process. So we're supposed to be building computational tools that strain designers can then use in order to build strains that have various biosustainability purposes for chemicals, natural products, and foods. So we've been doing that. We've been building these tools for a while. A lot of the tools today they were developed well before we were established as a group by other people, including the Paulson lab previously. But what we haven't done as much is to share these tools and educate others outside our research group on how to use these tools. So that's the purpose of these short courses. We're supposed to be stepping through these tools so that people who haven't done constraint-based modeling or haven't done sequence-based analysis, so on and so forth, have a concrete way to get their feet wet and learn exactly what we're doing and how we're doing it, to work with the software packages that we've been working with as well, and so that they can figure it out if these tools are going to be a good fit for their projects. In that said, it's also been apparent that we also want to be teaching these things also to new students in the Paulson lab and new postdocs who haven't seen these tools before, as well as any other collaborators who might be interested in them. So I think the scope of this short course will continue to grow, and we're just putting together these resources and soliciting feedback on how to present, what kind of material do we have too much of or too little of, that type of thing. So all kind of feedback is welcome. So the goals of the short course are first to increase familiarity of the concepts with each of the tools. The tool for today is constraint-based modeling. And these kinds of concepts are, for example, what kind of assumptions go into these models, or when is it appropriate to apply a constraint-based model versus a kinetic model. We want to be talking about things at a high level so you understand how these tools fit into a research project, right? So you can select the tools that are appropriate for your work. We also want to understand what's the utility of the tools. So what specifically are they able to do? So what kind of predictions can you make with one model versus another? Or how can they actually help you understand your data better? Like, what are the different algorithms available within a particular field in order to help your project? And then the last part is we don't want this to just be lectures where I'm just up here talking and describing these methods. There's tons of review papers. These things are well established in various fields. We also want to help you get your first hands-on experience with these tools so that you feel comfortable going and following up with this on your own. And of course, an hour or two of hands-on experience isn't really enough to learn it in and out, but hopefully through walking through the software installation process and initially running various aspects of the code, you at least feel more comfortable stepping into this, so you can carry this forward on your own. So that's the purpose of the course. In terms of the structure, this is the very loose intended structure. We'll see how much this sticks across these 4 sessions. But they're roughly split. Each session is split into 2 parts, one that has a data analysis theme and one that has a prediction theme, and each part has a lecture and an exercise. I don't know if we'll spend the full 4 hours today. It might end up being shorter, but we'll see. In terms of course resources, I'm trying to upload all of the files to this GitHub repository, which is public. So if you go to Module 1 FBA, you'll see the 2 exercises as well as the slide deck for today. And you'll see there's an IPython notebook and a model file, and so on and so forth. And you'll see here's the updated growth coupling suite. Is this the working version now, Tobias? Should I encourage everyone to download this and run this?
Yes, this should be the version. It's the same as in the other file, but as I said, there's the setup py, and then in the subdirectories the growth coupling suite as in the other archive.
Okay, great. Yeah, so please feel free to complete the installation homework that was sent out with that. If anyone hasn't seen this homework, please just put in the chat, maybe with your email, and maybe Tobias can forward that file to you before this is going, so that you can try to get this installed before these sessions start. Of course, you're completely, free to just watch us walk through the exercises. It's not going to be particularly boring to do just that, but to really get the full amount out of it, we want you to leave with like the software installed so you can carry this forward on your own. Yeah, which is this last point. Okay, any questions on the organization before we get started with the topic for today? Okay, great. All right, so to start it off, constraint-based reconstruction analysis, or what we call COBRA, one of the topics that the Paulson Lab is known for, and we teach a class on this, bioengineering, the bioengineering 212 series. Yeah, was there a question? Okay. So we teach a graduate class in the bioengineering department here on constraint-based reconstruction analysis. Dr. Paulson also published a textbook on it that I don't have the citation here, but there's a ton of literature out there if you want to have more formal background in this topic. But I'm going to be going through from the beginning a very broad overview of what is in a reconstruction and how do we build metabolic models from it. So to everyone who has been using these models, I apologize for the very basic nature of this work, but hopefully for people who haven't seen this or have been more on the data analysis side and less on the modeling side, maybe this is helpful. Okay, so the topics for this first session. What is a metabolic network reconstruction and how do we build one? What is the protocol for building one? And then I'll go into a number of specific issues for what makes these reconstructions what we consider high fidelity. And that comes down to accurately annotating your gene to protein relationships, gene protein reaction relationships, carefully handling your reaction mass and elemental balancing, as well as determining reaction thermodynamic feasibilities. All of these things make your metabolic models more physiologically realistic, and that's a key to getting accurate predictions at the genome scale, which is kind of the magic that constraint-based modeling is able to do. And then I'll talk about one of the key features of a constraint-based modeling, which is how we model growth, which is the central prediction of these models, via the biomass reaction, and describe how we set that up. And then last, describing how we can build tissue-specific reconstructions using gene expression data, and just an overview of some of the reconstruction tools. Okay, getting into it then. So just a brief starting out with the history of these metabolic reconstructions. Where did they come from? Well, in 1995, we started sequencing entire organisms. And this was really an eye-opening moment for a lot of people. I know Dr. Paulson himself mentioned that this kind of was shocking and really just sparked this entire field of systems biology, because for the first time, you could really know all of the content of an organism. Of course, we're still annotating the human genome, and we don't know what all these sequences do yet, but you could say it's within vision at that point. And so if you could say that you know all of the content, it should just be a matter of putting together the pieces in order to get an in silico representation of what an organism can do. And so that's what some of the early grad students in the Paulson lab did here at UCSD, Christoph Schilling and Jeremy Edwards. They used these early sequenced genomes to construct metabolic networks for some of the smaller organisms, as well as E. coli. And this was really the first high throughput in silico. This is in silico biology. This is really the advent of systems biology, when other people were starting to talk about network motifs and these concepts that didn't really exist or have much meaning prior to sequencing entire genomes. After this, after the early E. coli models, which were not comprehensive at that point, they were extended to other eukaryotes as well. For example, yeast by Jens Nielsen and others. And Jens Nielsen's lab has continued to work on yeast ever since, and has been developing kind of a parallel path of metabolic modeling to the Paulson Lab's E. coli work. Of course, you want to be, if you're doing systems biology, you want to be doing it in the human. And so there was a big effort between 2005 and 2007 to collect all of the available data on human metabolism. And of course, the annotation is not as good as E. coli, but the team in the lab was able to do this. I recognize 4 of these people: Monica, Nima, Inez, and Jan. And so they put together the first version of the human metabolic network, which is called Recon 1. And since there have been several other updates to that, the most recent being Recon 3D, which is called 3D because not only does it have the metabolic network, but they also mapped onto it all of the available protein structures and metabolite structures, and then integrated a structural analysis into the metabolic network reconstruction, which is something that we call a GEMPRO for a genome scale model with protein structures. And this type of structural effort is only going to be more and more powerful with the advent of AlphaFold and these machine learning technologies to improve our structural annotation. Meanwhile, a parallel effort was ongoing in E. coli, continuing to develop the E. coli reconstruction since this early model back in around the year 2000. So we've been continuing to add genes to the E. coli reconstruction model, and the latest version is IML 1515. The nomenclature for these in silico metabolic models for a long time has been I for in silico, and then the initials of the people who built the model. So in this case, it was Jonathan Monk and Colton Lloyd, so ML. And then the number of genes, metabolic genes in the reconstruction, so 1515 in this case. So we've been adding about 100 genes per reconstruction. And this effort also mapped on protein structures onto the E. coli network, as well as reconstructed strain-specific metabolic networks. So where you could see over time, we're not only expanding the metabolic networks, but we're also mapping on more and more data types to make these richer annotations of metabolism. And all of that should empower all this downstream analysis. If you're trying to interpret mutations, you can look at a protein structure now. If you see, some kind of strain-specific phenotype, now we have strain-specific models, for example. So all of this is just growing and growing. And hopefully these models will become useful for everyone's research. But let's take a step back and think a little bit about what actually went into these efforts. How were these reconstructions built, and why do people believe that these are accurate representations of metabolism? So in other words, how can you guarantee that your metabolic network is correct, is comprehensive, accounts for all of the available literature, so on and so forth? So what is in a metabolic network reconstruction? At the simplest level, it's just a list of reactions. So you can see here stoichiometry of glycolysis, so very well-known reactions here. And so this is, for example, I think in human. I'm not sure about that. I don't want to misspeak. But here we have not only the reaction, though, it really becomes a reconstruction when you start to link the reaction network, which you could find in Kagor in a biochemistry textbook, and you link it to the genes that can catalyze this reaction. So here we have a reaction abbreviation, we have the reaction stoichiometry, and then we have the genes that can catalyze it. And in certain cases, there are multiple genes that can catalyze it separately. So you have an OR relationship. Oh, here, maybe I could show it here. So here you have two different genes that can catalyze the same reaction, pyruvate kinase, and we call these isozymes. In other cases, you have two subunits that make up a complex that can catalyze a reaction. This we can have an AND relationship. So these kinds of logical and/or relationships are called gene-to-protein associations. And this is really where we're linking the content that we can read off the genome, right? Because we can look at the sequences of the genes and then turn it into a metabolic network, right? So these are the rules that enable that relationship. Now, we can also represent these stoichiometries mathematically with the stoichiometric matrix. So now this is a mathematical representation of all of these reactions. And so we have as rows the metabolites, and as columns the reactions. And you can see that you have a -1 when a metabolite is consumed, so on and so forth. And this is really the start of converting a network into a model that we can calculate with. And I'm not going to go into that right now, but I will discuss that in the second part. For now, let's just talk about the reconstruction. Okay, so this is glycolysis. Everyone knows that, you know, what glycolysis looks like. Of course, some organisms, you know, have the canonical glycolysis and some don't. But starting from a genome, how do you decide what your stoichiometric matrix for your organism should look like? Well, you start with your genome sequence, right? And from your genome sequence, you can read off by homology, homology to annotated, well-characterized genes, what metabolic capabilities, what metabolic genes you think are in your organism. And you call this a draft reconstruction. So entirely based on sequence homology, you could say, okay, this gene looks like PFK, so I think this organism has PFK. And you end up with a network that probably has some holes in it. It probably has some genes that are misannotated and don't have that function. And it probably has some genes that should be there and aren't there. And as a result, you have metabolic pathways that have a gap in them that are blocked in a sense and can't function as is. And so, so you can't just take this draft and assume that you have a working metabolic model. You have to curate it. And there's a variety of ways you can curate it, but it largely involves a lot of manual work, looking at these cases where there's blockages and trying to resolve them by looking for other homologous genes that fix those blockages. Or you can also look, for example, at known metabolic functions and say, okay, I know this has to be there and it's not there, resolve it like that. You can look at genetic data. So if someone has knocked out a gene and it removed the capability, for example, if you knocked out a gene and your cell can no longer grow on lysine, then maybe that gene is a lysine catabolism gene. And you can also look at biochemical data directly and fill in, for example, cofactor specificities. So isocyanated dehydrogenase, is it using NADP or NAD NADPH or NADH, right? A lot of times you have to go to the biochemical literature and hope that someone has characterized that enzyme very thoroughly. So with these kinds of steps, you can fill in these gaps and get your model to a point where it's as good as possible. And that brings you to testing. So you can test your model then, trying to predict growth rate or trying to predict gene essentiality, seeing whether your model can grow with certain reactions deleted, and whether that matches experimental observations of whether a cell can grow on a particular substrate or with certain reactions deleted. At this level, we're kind of testing the performance of the model, and I'll discuss how that goes. And once you have your curated model, finally, you can start to use it for prediction, which is what we'll discuss today, or to integrate various data types to try to make more sense of them. So you can integrate metabolomics data with your network and calculate hotspots of dynamic change and things like that. So that's the procedure, very broadly speaking. This has actually been very specifically formulated as a 96-step protocol, going from a draft reconstruction through a refinement phase, and then you convert into a mathematical formula. And then with that mathematical formulation, you can start to look at the model performance and see if it actually behaves like the cell does experimentally, physiologically. And if not, you kind of loop back and go through more curation until you're kind of happy with how it's working, and then you can release it. And we're going to step through a number of these steps now, but this is just to say that people who are trying to do a reconstruction for a new organism, there are very specific protocols that we've released on how to do this, and hopefully this is not the point of this is that we want reconstructions to have a standard of quality, right? That different groups don't have different procedures, different quality metrics, so on and so forth. So the community as a whole kind of agrees that these things need to be done to end up with a realistic representation of your metabolic network. And so as long as everyone follows this, the quality of models is generally pretty good, although there are some quirks that I'll describe later from different lineages. Well, I'll describe that in a bit. So when we're building these reconstructions, we often have these edge cases where you're not sure if a reaction should be there. And so we can annotate how confident we are that a reaction is actually there using these scores based on what type of data, what kind of evidence we have they're there. So this is just one example metric that we've used before, where we have scores 0 through 4. So 0 is there's basically nothing, no evidence that's there. 1 is that there's some kind of model-based evidence that it's there. For example, there's a gap that in a pathway that the model could fill with a reaction from a universal database. And it's like, okay, that could make this work, but you have no real evidence that it's there. So model-based evidence is around 1. Sequence-based evidence is if you have some kind of homology evidence that it's there, which is what a lot of the draft is based on. Or if there's some kind of physiological data that it's there, for example, some kind of transporter evidence or evidence that there's a metabolic activity around that gene. Then genetic data is above that, where you're actually knocking out a gene and showing that metabolic capability disappears when you knock it out. And then finally, at the best level is someone's actually isolated this gene and shown its chemical activity with a biochemical assay, which, for some organisms that's been done, and for other organisms hardly at all. And you're operating much more on this sequence and modeling level. So oftentimes with, in E. coli, we have a lot of this. And most of what we add is more of this. When someone's published a new study, we can add that to the model. When you're working in a new organism, you're often operating down here. and you're kind of stuck with that. That doesn't necessarily mean that your model isn't good. It just means that you should be cautious about the content of certain pathways around there. Okay, so let's step through a concrete example so you can kind of see how this actually goes. So this is this glucokinase gene, and so based on homology in your draft reconstruction, you might say, okay, there's a gene locus in the genome that looks like a glucokinase, and this glucokinase has an enzyme classification number 2712, and it has a reaction associated with it. That's great, but is it there or not? So then you might step back into the literature and then try to further curate it. So what does that actually mean? How do you further curate this? Why is this not enough? You have a gene and a reaction. Well, there's additional things that you can add. For example, this reaction is not proton balanced. So adding that is actually very important for reasons I'll describe, but basically because your electron transport chain is proton dependent, and you don't want to be just consuming and producing protons freely due to imbalanced reactions in your model. You can add how many subunits there are. You can add any kind of biochemical evidence that would bolster that this reaction is actually there, as well as reactions. And recently we've been adding charges, or we've been adding protein structures, metabolite structures to these reconstructions. But even prior to this, you can estimate whether a reaction, a metabolite will be neutral or not. So at a neutral pH, whatever the pH is within the compartment that this reaction takes place, you can calculate what the charge is on each compound and figure out whether or not you need to add protons to one side or the other to balance it. And so that's kind of how you transform this reaction to that reaction. Just one more comment on mass and charge balancing. It seems kind of trivial, but it's one of the main issues with these models. Is every now and then you'll have some poorly curated reaction in one of these models, and all of a sudden your cell will be producing infinite ATP. And you're like, what happened here? And oftentimes it's a reaction that should have been going the other direction, or a reaction that was not mass balanced, causing these types of things. And so on mass and charge balancing, it's mostly around protons, because most of the time carbons, oxygens, nitrogens are well accounted for. It's the question mark is almost always how many, or where do the protons belong? And just as an example for why this matters, think about your electron transport chain. So your ATP synthase is a proton pump powered machinery, right? And your cell has to produce a lot, has to consume a lot of redox equivalents, NADH, quinones, etc., to pump protons across the membrane in order to bring it back and produce ATP when you're growing aerobically. If you don't have a proton balance, you're either consuming protons every time you run a reaction, which is an energy drain, or you could be producing free protons, maybe in the wrong compartment, and essentially enabling your cell to have infinite ATP, right? So proton pumping is particularly important because of this interaction. Proton balancing is important because of its interaction with proton pumping, because protons can actually be metabolically active. And so, yeah, this is just another schematic of how this works. So you might initially just know what the names of your metabolites are, but you have to go to their formulas, and specifically their charged formulas, in which involves going to understanding what pH your compartment is, and then looking at the pKa, the acid dissociation constant of these metabolites, and then identifying what the major form is. And then whatever major form you select for these metabolites, you then set the stoichiometry of the reaction with balancing protons. And then this last part here, thermodynamic consideration, I'll talk about in a second. The question of whether the reaction is reversible or not. And then the question of localization is much more of an issue in eukaryotes, especially plants. I believe there's often a reaction taking place in multiple different compartments at the same time. So this can be quite difficult, and you have to look at things like signal peptides and any kind of fluorescent evidence. It can be quite messy. But this is the level you have to get to annotate or to construct a network for a eukaryote. For E. coli or bacteria with just cytosol, maybe a periplasm, here, it's an easier process. Okay, so this point of reaction reversibility. Essentially, when you have a reaction, it's very important to a constraint-based model to know if this reaction proceeds only in the forward direction, only in the reverse direction, or can proceed in either direction. In the ATP pump example, can your ATP synthase run in the opposite direction and produce ATP? In some organisms, it can. So it's very important to know in your organism. So how do you know that? Well, your reaction reversibility, every reaction technically is reversible for some concentrations, but that doesn't mean that your cell can ever run a reaction reversibly for the concentrations that it experiences. So the principle of reversibility is that the direction of a reaction is set by its delta G. It's Gibbs energy. And its Gibbs energy is essentially a comparison of the standard Gibbs energy, which is like an inherent favorability. How difficult is this reaction to run in the forward direction? And you compare that to what the concentrations are of the reactants versus the products. So even if you have a difficult to run reaction, if you have a ton of your substrates there and none of your product, your reaction will still proceed forward. In a test tube, that's very easy to organize, right? You can just put in your substrate and none of your product and wait for your reaction to proceed. In a cell, it always has both substrate and product present, so it's trickier to know whether or not a reaction is reversible in vivo. So most often, we're looking at literature evidence. Is there any evidence that a reaction or a pathway can run in the opposite direction? For example, arginine biosynthesis. We know that arginine can be biosynthesized from glutamate in a kind of an upward direction, from glutamate to arginine. We know it can run in that direction. Can it run in the opposite direction? Is it reversible physiologically? Well, in that kind of case, we would look to something like, can the cells grow on arginine? Can they actually catabolize it? And if they catabolize it, does it use the same pathway in reverse? Another very simple example is glycolysis. We know that glycolysis is mostly reversible because cells can grow on gluconeogenic substrates. However, there are some reactions like PFK that are not reversible because there's another reaction that bypasses these. So that's the kind of physiological evidence that you can go to. At a very mechanistic level, there are thermodynamic calculation methods where you can just directly calculate based on the concentrations that you know, or realistic concentration ranges, and delta G's that you can calculate these DGs, and that's the plot shown here. So these are these are estimated Gibbs energies for different reactions, and you can see that most of them are negative, meaning proceeding in the forward direction. A lot of them are around 0, meaning highly reversible. And if you would take into account uncertainty, you can actually, in high throughput, kind of estimate the reversibility of all the reactions in the network. It's not perfect, but it's a high throughput way. It's, again, you could think of it like a draft reconstruction of your reversibility. Okay, so that's the last piece in constructing a high confidence metabolic model. If you have high quality GPRs, high quality mass and charge balancing and elemental balancing, and high quality reaction reversibilities, you're in pretty good shape. The other piece of a reconstruction that's really important, though, is how we model growth specifically, because this is the key prediction with a metabolic model that we're often interested in, is how fast can it grow? Will it grow with a particular knockout? What is the trade-off between growth and production of a compound that you're interested in? Growth predictions are essential and central to metabolic models. And the way that these models predict growth is a little bit different than the rest of the network, because it's not a real reaction. We actually construct an artificial reaction called the biomass reaction that accounts for the drain of all biomass precursors from the cell. So in a sense, it's a lumped reaction modeling. We know that whenever the cell divides, it's diluting out its pool of macromolecules. Every time it divides, it effectively halves its DNA and RNA, halves its lipids, halves its proteins, and it has to resynthesize these things. So the rate that it can resynthesize these things is its growth rate. It's its growth limited rate, right? It can't grow any faster than it synthesizes the precursors necessary for the macromolecules. And so we kind of work backwards from this point and say, okay, we know that these are the components, and we know these are the precursors. We calculate how many of these precursors, and we make a stoichiometric equation that is the drain of all precursors on a millimole per gram dry weight basis. And then the flux of this reaction is directly interpretable as a growth rate, because the units of flux in our network are millimole per gram dry weight per hour. So because this reaction has units of millimole per gram dry weight, the flux has the units per hour. And so it's directly a growth rate. It's kind of an interesting trick. So how do we figure out these percentages? Well, you actually have to go in and measure them. So people have measured the protein content of different cells. This is just an average, you could think of it. Protein around 55%, RNA around 20%, DNA 3%, so on and so forth. And then for example, for DNA, how do you tell how much is CTGA? Well, you can just read it off the genome, right? In RNA, you can do something similar, looking at expression data. And in protein, you can do something similar as well, or you can just look at mass fractions that have been measured. And a lot of these have been measured. So this type of biomass characterization enables you to parameterize the coefficients in your biomass equation, which then enables you to model growth. And that's kind of the last piece of a metabolic reconstruction, is the construction of your organism-specific biomass function. And this could be quite different across organisms. There's cyanobacteria that accumulate large fractions of lipids. RNA fraction can vary with growth rate, and people have studied how much this affects your model predictions. The answer is not a whole lot, but a reasonable amount. So on and so forth. And then the last piece that I want to mention is, so we're talking about organisms, right? Or like this is the E. coli metabolic network. This is the human metabolic network, right? But we know that within a macromolecular organism like the human, the network isn't the same in every cell type, right? And even within E. coli, your metabolic network, the expressed proteins, the proteins that are actually there in catalyzing activity, changes based on condition, right? Sometimes you'll turn on a gene, or you'll turn off anaerobic genes when you're lacking oxygen, or so on and so forth. So so how do we account for that? Well, the main method has been to use omics data like gene expression, and start with your fully curated, larger whole organism network. And then based on the omics data for a particular condition or for a particular cell type, you extract out the active network. And so you can see a lot of this inactive part has been grayed out for this particular condition. And then you can end up with a condition-specific predictions or an organism-specific model. There's two key features here. One is it's not always apparent what these cutoffs are for adding a gene or not. So there's yet another kind of curation process when you're trying to say, should this pathway be in my cell under this condition or in this organ or not? The other piece is that these networks are not set in stone, right? So what if your cell shifts conditions slightly? Do you want to build a whole other metabolic network for that new condition or not, right? So you have to take into account how rigid this condition-specific model is to determine whether or not it's worth building one. So this has been a challenging area, building these condition-specific models from organism-specific reconstructions for quite a long time, and there have been various algorithms for doing that. Just to close out this session then, on reconstructions, there's a variety of different reconstruction tools that have been published. The Raven Toolbox, for example, in MATLAB is a big one. Seed and KBase have been building tools. We started out with Symphony, which was developed by Evelyn, who's in our lab now. And we've even just used Excel tables. So there's a lot of different tools, and they can each handle different parts of this workflow in an automated way. And if you're interested in building a reconstruction, I wouldn't point you at any specific tool. I would just say maybe examine the different tools and see what you like working with. Okay, so to just close out the session then, I want to talk a couple of applications of reconstructions for data analysis. And the first of these is what do we learn from comparing metabolic networks across organisms or across reconstructions? And specifically, what do we learn about phylogeny, about the diversification of metabolism from an evolutionary perspective? Well, one effort at doing this, we published in 2014, was to compare with a PCA, a principal component-like analysis called multiple correspondence analysis, all of the available curated reconstructions. And what we're seeing here are the two dimensions that separate these reconstructions the best based on their reaction content. And you see some interesting trends here. For example, you could see that you could see a progression over time of the E. coli models, the lineage of E. coli models from this original 2003 model up to at the time the 2011 IJO 1366 model. IMO 1515 wasn't out at this time. But you can also see that there's another cluster over here, and then some clusters in different areas. And part of the interesting finding here was that they don't always separate based on organism or based on metabolic content. Sometimes they separate based on the fact that some of these reconstructions were made by starting with like an E. coli reconstruction and mapping it over to your organism of interest, right? So they were made by homology to metabolic models. And as a result, the resulting organism-specific models still kind of look like the E. coli model, which is kind of a curious feature that I don't know is really intended or desired of these features. Of these metabolic networks. So if we remove this bias, right, and not think about how these models were generated, we can also just look at the KEGG gene content. So this is just a very blanket analysis of metabolic content in KEGG for these organisms with the same MCA type analysis. And here we see a very distinctive or more distinctive separation based on phylogeny. So you can see the eukaryote all grouped together with some kind of variance here, and then bacteria in archaea also separate. And if you look at what defines this factor, it's a lot of eukaryotic specific machinery, such as transcription and translation processes that are different, and signaling pathways that only exist in eukarya. Then if you try to break this block down further, you can see that there's yet another separation between bacteria and archaea within the smaller cluster. And once again, you see that this dimension is defined by archaea-specific processes like archaea have a very special type of membrane, and the metabolic genes that make that membrane are a differentiating factor between these groups. So you can learn a lot just by this kind of phylogenetic metabolic network analysis, and that's something that we've continued to explore. For example, to analyze pangenomes, or how does the metabolic content differ across strains of the same organism. Now we're looking here at different phylogroups of E. coli with a very similar type of analysis. And once again, you can see that there is clustering in the sense that, in the way you would expect. You can see the S group is over here, B1's over here, B2's over here, so on and so forth. And then you can interpret these weightings to try to understand what is actually metabolically different about these groups that have been defined based on physiological methods. So what does this matter? What, like, who cares? Like, like, why is this helpful to a scientific research project, or is it just general curiosity? Well, let me just give you one quick example. So this was done by looking at gut microbacteria, and some of these bacteria were classified as pathogenic. And they looked at, this was done by Xin Fan, a graduate student in our lab. She looked at the metabolic gene content across these organisms, and she noticed that there's this tagatose-related metabolism that only showed up in this particular phylogroup that was associated with pathogenicity, which is this block here. And so this could suggest this tagatose is derived from intestinal mucus, and this could suggest that these organisms are, because of this metabolic capability, are better able at surviving in a particular niche where these metabolites are present within your gut. And so by understanding these large-scale metabolic differences, you can actually infer a lot about the function of these organisms. So I just want to highlight this as one way you can actually do data analysis just by looking at the metabolic network, even without accounting for any other data at all, just thinking about the metabolic network itself as data. But of course, we often do have other data, gene expression data, metabolomics data, and so we want to we also want to use the metabolic network to analyze that. And the method of choice for a long time has been to look at metabolic pathways. You might be familiar with gene set enrichment analysis, where the pathways are, it's a pathway enrichment tool, where these pathways or gene sets are defined manually through manually curated sets, largely at least, through this MSig DB. You can see some of the example categories of gene sets here. And whenever you have a new gene expression data set, you can say, okay, these particular functions were differentially expressed. And you know, it's a nice way of getting it. But the question is, do metabolic models offer a different set of genes, and maybe a different viewpoint than what you would get from manually curated sets? And so the answer is yes. You can actually calculate pathways from a metabolic model using this type of formulation called a min-span algorithm. There's other pathway calculation methods out there. I don't want to go into them too much. But basically, you take your stoichiometric matrix I defined earlier, and you can calculate pathways from a sparsified null space of this matrix here, which is some linear algebra for you that is maybe not worth going into here, but the visual representation of this is maybe more helpful. So if you have a metabolic network that looks something like this, a pathway breakdown of this network might look something like what's shown on the right, where you have different routes through your network that are calculated as pathways because they can operate at steady state on their own. They're independently operating modules in that they run at steady state without needing any kind of material from the rest of the parts of the network. So this network has one pathway here. You can see one pathway here. one that's just a loop here, and then one alternative route here. And the sum of these reproduces everything that your original network can do. But this has maximum sparsity. And why is sparsity interesting? Well, because it increases your interpretability. And it also, sparsity is just a biological principle that underlies, or you could say efficiency is a principle that underlies metabolic networks, right? There's no reason to have 20 reactions when 3 reactions would suffice. And so these pathways often end up to be very similar to the canonical pathways. But these pathways enable us to do a lot of different things. For example, you can calculate pathways that you didn't even know were physiologically meaningful. So these manually curated pathways might not think that shikimate to tryptophan is a meaningful pathway, right? Like you might be able to find some route there, but there's no reason to think that it's doing anything within the cell. But this minspan algorithm calculates this as one of its vectors. And if you look at gene expression regulation data, it actually turns out that all of these genes are repressed by the same regulator, which would suggest that this is intended as this has an intended physiological role. Otherwise, why would you regulate it all the same? You can also identify dependencies between core metabolic functions and functions that are supportive to those core metabolic functions. So this is IMP biosynthesis, and it's known that this is going to be regulated by pure R. But what's interesting is there's a tetrahydrofolate cofactor regeneration step that is also regulated by pure R that minspan picks out as being part of this pathway. So normally, the canonical definition of pathways would say, okay, this is folate, this isn't IMP biosynthesis, but minspan picks out that it's essential, and E. coli regulation agrees that this is all one module together. Just in the interest of wrapping this up, you can also map other data types, for example, macromolecular associations, protein-protein interactions onto these pathways, and it turns out that these pathways are predictive of whether or not there will be protein-protein interactions or genetic interactions, as in epistasis. So if you delete one gene and then you delete another, how often is it synthetically lethal or detrimental, some kind of interaction? These kinds of calculated pathways perform better than curated networks at predicting these interactions. So in other words, it seems even though that we've had these pathway definitions for a long time, the metabolic model often knows more about what is actually important for function than the well-established curated gene sets that have been around for a long time. And therefore, metabolic networks should be considered as a valuable resource for data analysis on top of what is out there. Okay, so I finished up just in time for this session. Thanks for sticking through that if you already knew everything about metabolic models, but maybe there's a paper or two there that you hadn't seen before. Just to summarize briefly, these reconstructions are structured knowledge bases. We put together all the knowledge that we have for an organism, and we try to make it we try to interrelate them together so that we have gene-protein reaction relationships, maybe we have protein structures, we have reversibility. It's all in one place. And then we, in a minute, I'll talk about how we convert that into a mathematical model for prediction. The reconstruction itself contains data types, and we can actually analyze those data to understand more about phylogeny and other things as well. And then we can also map on gene and protein expression onto pathways, and the pathways provided by metabolic models can often provide even more accurate predictions than user-curated models, which is pretty interesting. Okay, so that's it for part one. So there's 2 exercises. Let me take any questions or any comments on this before I move on. Okay, if not, then let me just get into the first 2 exercises. So The first exercise is something everyone can just do web-based. It's very straightforward. If you have not used Escher, it's one of the best tools out there right now for getting familiar with metabolic networks and metabolic network reconstructions. So this is the website for it. You can go there now. It's free to use. You don't need an account or anything. But I'm just going to be stepping through this. And I encourage you all to, if you're not familiar with Escher, step through these steps and explore the documents, because this is going to be the best way to learn more about network reconstructions, as well as just metabolism in general. So whenever someone's starting out a new project, I always encourage them to build a metabolic map for their network so that they can really get familiar with the metabolism of their organism. Okay, so what is Escher? It's a web-based platform. You can select organism, you can select a network to view, and you select an underlying metabolic network reconstruction. Okay, so here I'm just going to select central metabolism, which is the IJO model, and I'm going to load a map. Okay, so when you load a map, you can see here that there's central metabolism that you might be familiar with. You have glycolysis, glucose phosphotransferase system, PGI, PFK, so on and so forth. If you mouse over these, you can actually open up information about this reaction in the big database here and learn, okay, this is the reaction, this is all the models that it's a part of. There is a, well, yeah, you have to go to an actual model to see the GPR for it. So if you click on a model, you can see what genes can catalyze it. So this is this GPR rule. So in this case, it's this or this, a subsystem, some default bounds in this particular model, so on and so forth. And then you can see a variety of external database links as well if you want to see the EC number for this, or a MetaNet X reference here. All of this can be found. Okay. So what's the point of this, right? Well, it's just a viewer at the first level, but there's a couple things you can do. First, you can not only, well, okay, let me show you a couple of the features. First, it's not a static map, it's a dynamic map. You can highlight things and move them around. You can annotate text, so I can edit this text. Transhydrogenase proton pumpkin. It's all live, and you can even, because it has a metabolic network behind it, I can add reactions. So if I'm interested in seeing what else is this reaction part of, I can add this reaction, and I can see, okay, it's this metabolite is coming from gluconate also. Where does gluconate come from? Here, whatever. And now I have gluconate transport in my model, right? So so that's part one. You can actually build models from scratch. I could have started from no model, but I just want to show you different things. You can do searches, so I can look for protons, HC, and unfortunately, it's all over the place because it's in different things. And then the other thing you can do is actually load data onto here. So you can load gene, reaction, metabolite data, and it will color reactions, nodes, and highlight things. You can, so Tobias in his demonstration later in this session will show an example of loading flux data onto an Escher map so you can look at a solution. Okay, so I would encourage everyone to look at this. You can load your own model here. You're not restricted to the models that they have. And then you can also load your map. So you can save or load your map or export it as a PNG, so on and so forth. So for anyone getting started with metabolic networks, Escher is the best place to begin. Before this existed, we used Symphony, and we had gigantic maps. We still don't have all of the E. coli model in one map. It's still a little laggy, but we've we've built quite a few of these. Okay, so that is part one. I would encourage everyone to explore this tool if you're not already. Part two is Escher FBA, which is an app for Escher. So Escher has a number of Escher apps like Escher FBA. It also has Escher with structures. And so this just has metabolite structures and Escher with tooltips. For a while, there was a lot of active development with building these demos, but these are kind of all just hanging out now. But I guess my point is all this code is available and encourages active development. So if you're really interested in Escher, you could even build your own demo for it. But taking a look at one particular demo that's of interest is Escher FBA, which is one of the nicer, more intuitive ways to understand flux balance analysis, which is the metabolic modeling that we can do with the metabolic network reconstruction. And so I want to get back to this in a little bit. So here I have a question. So the rest of the lecture, the part two of the lecture, is on flux balance analysis and how do we actually do constraint-based modeling. Now, there's a little bit of a non sequitur here where I actually have a coding workflow before the lecture here. How do people feel? Would you rather sit through another 40 minutes of lecture before we get started, or would you rather just execute this task and then go through the lecture just to kind of break up the monotony? What do people vote? You can just put it in the chat. Okay, I'm just going to go through the exercise then. Okay, so let's then step into the exercise first in FBA. So the first exercise for the day is a prelude to the more advanced Cobra modeling that we're going to be doing later as part of Tobias's growth coupling algorithm. And that is to do to go through the CobraPy, which is a Python-based software package for constraint-based metabolic modeling. Okay, so just so everyone can follow along, the location of this is growth coupling workflows. Oh, sorry, just take a step back. GDGA short course module 1 FBA, part 1 exercise, and then FBA introduction. Okay, so that's where we're going to be stepping through here. Okay, so just as background, CobraPy has a read the docs at this location. Maybe I should put that in the slides. And this workflow is largely derived from this read the docs, but we'll be stepping through the different components of a metabolic model as well as elementary FBA. Okay, so to start off, we can import the CobraPy package and then read in this model IML1515.xml, which is the latest E. coli reconstruction that we've been talking about. And we can begin to explore the properties of this model. Okay, so we can see here even a memory address for this, but we can see it has 1,877 metabolites, 2,712 reactions. We can also see that we can see its objective here and any available compartments. So that's at the very highest level what is in this reconstruction. Now we can start to look at more detailed lookups, either by index. So just looking at the first index, we could say, okay, what is the first reaction? ID, stoichiometry, lower and upper bound, and print this out. Or you can also look it up if you're interested in a particular reaction. You can use the getters, get by ID, and also find the same information there. You can also use this to change constraints. So if you get by ID, it's not just viewing. You can also change the values by this method by just setting them, okay? So at the very basic level, that's all we want to do, is just start to explore what is in a metabolic model and get familiar with the structure so that when we're coding with it, we know what is where and how to access it. But we can also, one of the more common tasks for metabolic modeling is to actually add reactions to a model. So here we have a reaction, well, it doesn't really matter, but it's a cell biosynthesis, but we have to define a reaction name, abbreviation, name, subsystem, and bounds. And then we have to define the information on all of the metabolites in the reaction if they're not new metabolites. So for example, in the case of a proton, it was already in the model, so we don't have to give it all the information. But we do anyways, just for clarity here. And so we create metabolite objects with the name, formula, abbreviation, formula, name, and compartment. And then finally, once we have all of all our metabolites, we can add them as metabolites, basically defining the stoichiometry of this reaction, and we end up with our new reaction. So this is reaction.reaction we're printing out. And we also need to define a GPR rule if we want any of the gene specific functions to work. So here we just have this information, and then we can add in this reaction into our model with model.addReactions. Okay, so if you're adding a heterologous pathway, this is what you want to do. Now we can step into flux balance analysis. Okay, so should we take a break maybe for a little bit? Here, why don't we take 5 minutes and then come back, and is that fine, Tobias?
I would recommend this, yeah.
Yeah, I just feel like I've been talking for a long time. Okay, so here, so in your own time, explore Escher and explore this notebook, and then I'll come back and we can do a more detailed discussion, and I'll field any questions on this, okay? And also, if there's any installation troubles still, please put them in the chat and we can try to resolve them because I think we'll have plenty of time in this session. All right, thanks everyone. Sorry for talking your ear off. We'll be back at 9:18. Let me put that in the chat.
Thank you.
Thanks, Dan.
Great. See you in 5 minutes.
see. Okay. Welcome back. Sorry for talking your ears off without a break. I think we'll probably try to have another break before Tobias's session, maybe. Okay, so continuing on then, just to reiterate, so far all we've done is import our metabolic model, which is the E. coli model, and shown how to inspect different parts of the model, as well as change them, and then add reactions, which is a very common task for metabolic networks. We're trying to either curate them or often add heterologous reactions. But now, without really going into the details of what we're doing, let's start to simulate them. So we talked already about this biomass reaction, biomass subjective function. And here we have the E. coli core biomass, which is what's used by the IML 1515 reconstruction right now. And we can simulate this model, and that's what we did now. We've only printed out the objective just to show that this is what was being maximized, this biomass reaction. And we see that we created this solution variable, which has 4 key attributes. The first is the objective value. So we already described that when we're using biomass as the objective, that means a growth rate. So we've calculated a growth rate in the objective value. It also returns a status. So sometimes a model can be infeasible, or there can be various issues. And so it can throw a status other than 1 is usually what's used for a successful solution of the problem. And then the reaction fluxes. So this is, it's not just one flux that we simulated all of the fluxes throughout the network. So these are the reaction fluxes that enabled this growth rate. And so this is a Panda series that is indexed by reaction identifier. So you can actually look up the fluxes by reaction name. And then the other piece that is maybe less familiar to people is what's called the shadow prices. In linear and convex analysis terms, these are called the dual variables. But they actually have a really interesting interpretation. So whereas your fluxes are values for every reaction, we talked about how they have units of millimole per gram dry weight per hour. So they have an interpretation, very physical interpretation. Your shadow prices have the dimensions of the metabolites. So if this is N, if there's N reactions, there's N fluxes. If there's M metabolites, there's M shadow prices. And these shadow prices are how much more of your objective value would you get from injecting more of that particular metabolite. So it's a sensitivity. It's the sensitivity of your objective to every metabolite. Now that's a useful thing actually for a couple reasons. The easiest interpretation of that is that it's what is your model limiting? Like what is limiting further growth or further production in your model? So for example, if you're oxygen limited, then oxygen will have a high shadow price. And oftentimes you can use this to debug your model, because sometimes your model won't grow because maybe you forgot to provide it a nutrient or something. And you can just look at your shadow prices. As long as the model is feasible, you can look at your shadow prices and actually debug what is wrong with your model. You could say, oh, it's missing this component or that component. Then you can add it as an exchange reaction. But yeah, typically for FBA, we're talking about the objective value, which is most often growth, and the reaction fluxes. Sometimes we change the objective value. So here we can see what is our objective. It's 0.876, which is a very realistic growth rate for E. coli growing on glucose aerobically. So that's a nice realistic value. Okay, so this is our growth rate of our in silico cell, 0.876, and it's in the solution dot objective value. Now we can also see a really nice view of this. CobraPy has this function called summary, where we see not only the objective value that we just calculated there, but also all of the metabolites that are being exchanged by the model. So you can see here, these are the abbreviation _E means that it's extracellular. So these are external metabolites, and they're exchanged. This is an exchange reaction, which is the interaction of the cell with the environment. And the fact that these have a flux means that the model is taking them up. So what is it taking up? Largely metals, calcium, chloride, cobalt, all these trace metals, as well as glucose, which is its main carbon source. So it has C number 6, and it's 100% of your carbon flux, as well as oxygen, which is taking up at 22, which is quite high, and some phosphate, some ammonia. And what is it secreting then? Well, largely CO2. So you can see the by carbon mole, it's producing 99.99% CO2, which indicates that this is a highly oxidative growth state. It's not producing any acetate or anything else. So this is a very nice snapshot of what your cell is doing metabolically. And oftentimes we want to change these. Like all we want to do is change these exchanges and see how these things change, right? That's often with metabolic modeling all we want to do. So this is an overview of the exchanges or what is the cell taking up and secreting. We can also do an overview of individual metabolites and see what reactions are exchanging them. So this is NADH, for example. And you can see as a percentage of all reactions involving NADH, which reactions have the most flux. So you can see GAPD, which is glycolysis, has 40%. That's expected. Pyruvate dehydrogenase, malate dehydrogenase, alpha-ketoglutarate dehydrogenase, all TCA cycle make up the majority of the rest. So these are dominating the NADPH production. And what is dominating NADH consumption? I would assume it's the electron transport chain. So if we look at this, yeah, it's 90% the NADH dehydrogenase. So all of it is going to pump protons in order to power ATP production, or 90% of it is. Some of it is producing FAD and a variety of other, maybe lipid synthesis metabolism. So this is another really nice snapshot way to understand what's going on in your network. Recently, we're interested in carbon dioxide, for example, in some of the production strains. So we can look at all those reactions through this lens. Okay, so that's a very baseline growth state. The model came with all of these exchange flux values, and you calculated growth for that, which is effectively glucose aerobic. What if we want to change that, right? So what if we want to do something else? Well, one very simple thing we can do is change to an anaerobic condition. So to do that, we change the lower bound on our oxygen exchange to 0. So it can no longer take up oxygen, and we can just redo a simulation there. And you can see our growth rate now has decreased all the way from 0.87 to 0.157 from doing this, right? So very clearly a very major change, and we could repeat all of this and see how the metabolic nodes have changed, what pathways picked up the slack or might have changed due to a shift from aerobic to anaerobic metabolism. The other very common thing that we want to do is to knock out a gene or knock out a reaction, right? So this is often done to design cells, as Tobias will talk about. Or to compare model results to experimental interventions to see if the model is accurately predicting metabolism, right? Can it predict essentiality correctly or not? So we can do that here with CobraPy built this knockout function. You could also just do it by setting the bounds to 0, but there's a special function for it, so why not use it? And you can see in this particular case, I'm knocking out this glycolytic enzyme PGI. We're still anaerobic because we changed our oxygen to 0. And you can see our growth rate has gone from 0.158 here to 136. So it hurt growth a little bit, but not a whole lot, right? So you could do this type of analysis. There are also tools to just do single or double gene deletions on every gene. So you can just see it takes a little longer because it's running multiple different simulations. Here I'm only doing 20 genes, but still it takes a little while to get going. So just wait for that. But typically, this is what you'll do to test the performance of a model against experimental data. You'll knock out genes in experimentally, and then you'll knock them out in the model. And you'll see how many times does the model predict growth or no growth versus the experiment. And so you can see here, knocking out this gene resulted in no growth. Whereas this gene did nothing. And so we have a number of essential genes here. I don't know if they're conditionally essential, if they're only essential on anaerobic metabolism, or if they're always essential, but you could try a couple different conditions and extract this information, do this type of thing. Then the last very common thing we'll do with the metabolic model is to change the objective. And so sometimes we're not interested in growth. Sometimes we want to constrain growth to a high level because we measure it, and we want to optimize production of something instead. We want to look at yield, or we want to understand how much can it produce of something. So here I'm just showing an example. You just change model.objective to whatever you want to, and here you can see that acetate, its maximum flux is 25.256, which is quite high. Okay, so that's the last part here. So that's all of the basic FBA functions that are very common to do. A couple other types of constraint-based modeling that are pretty common two of them are around well, they're all around further characterizing your solution space, your space of possible flux values. The first is flux variability. So flux balance analysis, we optimize a single objective like growth or production or something, and but the reality is that the fluxes that we get back are not unique. The objective value is unique, but the ways to achieve that objective value are non-unique. You can have parallel paths, and your model doesn't know which one to pick. It'll just pick a random one based on how the solver works. And so your fluxes will have uncertainty. And there's a couple different methods to characterize that uncertainty, the first of which is flux variability analysis. Now, flux variability analysis, you'll often constrain your first objective to like 90% of its maximum or something. And then you run a series of subsequent optimizations, maximizing and minimizing every flux in that network one by one, in order to say, okay, how wide are the bounds on these different reactions, right? Which reactions are constrained to be at a certain level, and which do I have a high degree of uncertainty still? And that can tell you how much two things. One, how much do you know about certain pathways so that you don't make a judgment call that, oh, this is definitely going to be high activity if there's another solution that maybe is low activity. And two, you know which reactions are constrained to be high, which are going to be the most critical variables for critical pathways for sustaining your objective. And so you can run flux variability analysis here, which takes a little while. So I'm only run it on a handful of reactions as example, so you're not sitting and waiting there forever. And you can see the minimum, the maximum fluxes calculated here. There's also a nice summary that they can spit out where you get different ranges on exchanges, which is gives you some uncertainty on your exchanges themselves. And then, yeah, you can also do the same thing on metabolites. I didn't actually execute this, but you'll just have to run that. Okay, so that's the first way to characterize uncertainty. It's just by maximizing and minimizing every flux in your network. That's called flux variability analysis. The second way to characterize uncertainty is by what we call convex sampling. And so in sampling, we're not running an optimization at all. Instead, we're just grabbing random points within our solution space. Now, it's not easy to grab random points, so you need a special algorithm for it called Markov chain Monte Carlo sampling. But it essentially just, if you have a cube, it just gives you a bunch of points within that cube. And that's another way of characterizing your uncertainty, right? Because you can look at then at distributions of those reactions. And you can see here, we can run this sampling method, and you see I have a series of samples here. And so TKT1, you can see, has different flux values, and you can characterize its uncertainty by looking at how much that actually can differ, right? Sampling is a highly parallelizable process. So because you're getting a bunch of sample points, each one is calculated independently of the others. So you can use more processors and run this faster. So I would just pay attention if sampling is taking a long time, make sure you're exploiting all of the options in the sampling code. And there's a couple different sampling algorithms that CobraPy supports. There's the opt-GP sampler, and then there's an artificially centered hit and run sampler. There's basically a trade-off between time and accuracy there. So so sampling is another excellent tool. And then the last tool I want to highlight in this demonstration is production envelopes, which is something that we're going to get back to with Tobias's example as well, looking at production versus growth in growth-coupled solutions. So a production envelope is essentially a phase plane between two variables, where you can look at interactions between them, and you can calculate them explicitly with CobraPy. They've actually deprecated some of their capabilities here, so it's not as nice as it used to be, but you can see here that we can calculate something like oxygen uptake versus carbon yield. So there's actually kind of a sweet spot where your carbon yield is the highest, and then you're taking up more oxygen than you really even want to take up, so you actually get less efficient. So yeah, you can look at key variables of interest. Maybe you're interested in substrate uptake versus production, growth versus oxygen, whatever you're interested in, you can study with these types of things, and that gives you more of a behavior of your system. Okay, so that's the introduction to CobraPy. The last thing I want to say is, if you're more of a visual learner, Escher again is your friend. It's not easy to know what these reactions are. Even these nice summaries that tell you all the reactions involved. You might want to build a pathway for this. But there's actually an FBA version of Escher that I was starting to go through here. And this is an Escher map that actually runs an FBA simulation behind the scenes, so live. So here you see this, it actually calculated the E. coli biomass at 0.87, which is that same value we calculated with CobraPy. And you're visualizing the Flux solution that results in that on this metabolic map, right? So, and you can, not only that, this is not just a static simulation, you can even constrain this, so I can even knock out this reaction. So we talked about how you can do a knockout in CobraPy. I can do a knockout here, and the flux reroutes, and I can take a look to see if it affected my growth. It went from 0.87 to 0.86. So it affected a little bit, but not too much. But what if I keep knocking things out? Okay, now I'm down to 0. So yeah, it doesn't do anything now. So I accidentally killed myself. But if you're interested in a more intuitive way to learn FBA and understand these things, this is a really nice tool when you're just getting started out, to understand the effect of knockouts. You can of course expand this network. It's fully functional Escher, so you can build your own pathway map here and then play around with knockouts and designs and things like that. So this is the last tool that I wanted to highlight as kind of the basics of FBA, getting started and getting more familiar and more comfortable with constraint-based modeling and metabolic network reconstructions in general. Okay, so part 2 of me talking your ear off. Any questions or anything before I get back to the lectures? And again, if you were, actually, I'm interested if you were able to install the software at this point, because as we're heading into Tobias' section, I'm curious if all these issues were resolved, but if anyone was able to, I'd also be interested in hearing that. So feel free to put it in the chat if you're up and running with these different tools. If not, just to highlight once again, they're all available on this public GitHub. Oh, great. So it's working for people. That's great. So this is all working, and again, the intention is that this isn't just demo code. This is something that you can just immediately apply to your own work. Like this is CobraPy. So so all of this code is stuff that you can take snippets out of and apply to your own work. Tobias will show some code for Escher integration within CobraPy. So hopefully all of that becomes accessible, or more accessible if you ever felt like it wasn't. Okay, so that is demo 1. So I'm actually going to go back and now cover the theory behind all of these models for another 20 slides or so before we get into Tobias's demonstration on growth coupling. So part 2, so just in terms of a layout, I think we'll do these slides. I'll try to keep it to like half an hour or so. We'll take another break, and then we'll do Tobias's demonstration, and it looks like we might finish up by hour 3 or so. Okay, so we've already talked about metabolic networks, and we've already run through a workflow for simulating with them, right? But we really haven't talked about how that actually works. So part 2 of this is, how do we turn a network, a static network, a static reconstruction, into a metabolic model that we can simulate and predict with, right? So the best analogy I've heard for this is traffic flow. So you can look at a map, and it actually looks a lot like a metabolic map. You have a bunch of nodes that are interconnected, different streets, different, you think of those pathways, so on and so forth, but you don't really know where Flux is going, where the cars are going, right? It's just static. But once you actually look at the traffic flow, then you get an idea of the intention of the city, of the cell. Where is it trying to go? What are the high flux and low flux pathways, right? And so we're doing the same transition here from a static metabolic network into a calculation of flow through our network based on the objectives of the cell, right? So the objectives of traffic are largely get from home to work, right, and back. And so, you could understand the design of a city just by looking at traffic flow. We can do the same thing with a cell. We can understand the organization of a cell by looking at the flow of material through. So this is exactly what I just showed with Escher FBA. You can actually calculate flow through one of these networks, and you'll find the same thing. This is one of the earliest findings in systems biology, was that there's this high flux backbone, that glycolysis is order of magnitudes higher flux than other parts of the network, right? And there's a reason for that, because your cell needs so much more energy than it needs anything else, and also because all of the precursors to make everything else come from these same pathways. So it has this dual role in biosynthesis and energy production, right? So we've already talked about how we can convert our metabolic network into a mathematical form via the stoichiometric matrix, right? So just to reiterate, our metabolites are rows, our reactions are columns, and you can see that the stoichiometry is the stoichiometry within this reaction. So -1 ATP is getting consumed, so that all works out, right? But how does this actually enable us to simulate? So we have a matrix, right? How do we get flow from this matrix? Well, that brings in the mass balance constraint. So a mass balance equation is universally true in a well-mixed cell where you don't have diffusion. And that is that the change in amount of material is production minus consumption, right? If you produce more, you get more. If you produce more than you consume, you get more, and you have a positive derivative here, right? But for constraint-based modeling, we the key, no, I guess the key is that we actually consider our cell not dynamically with changing metabolite concentrations, but we consider it steady state. We assume that everything under the current state, because our cell is happily growing with no dynamic perturbations, on average, you know, like, yes, it divides and there are dynamic processes going on, right? It's not motionless, but on average, it's not changing its state. So this dx dt derivative of metabolite concentrations is 0. And so we have a SV equals 0, which is like, if we would normally solve this with a, you know, this is a dynamic equation. We'd solve this with differential equations and solve this, you know, as a time series over time. We actually use a different set of tools called convex analysis, which is rooted in linear algebra, and treat this as a set of matrix constraints instead. So SV equals 0 is a set of linear constraints. What does a linear constraint look like? Well, it looks like a line in flux space. So our variables here are the fluxes, and the columns of S look like these lines, right? So it actually forms a cone within flux space. And every point within this cone is a flux state that satisfies this equation, meaning it's a steady state flux solution, okay? The other piece is that I should mention is that this is actually SV equals 0 is unbounded. It's like, yes, this is a blue space, but it's an infinite blue space. You have to put some kind of bounds on your fluxes, and so that's where that's where two things enter. One, for internal reactions where we don't know what the real max and min of this reaction is. We bound them based on reversibility. So if they're irreversible, we say 0 lower bound. Otherwise, we say they're infinite. So effectively infinite. So like plus minus 1,000 is high enough that it's considered infinite. However, for exchange reactions where we actually can measure them, for example, glucose uptake, growth rate, exchange secretion rates, we can use that data and set a V constraint on those values, and that actually bounds this space so that it's no longer infinite, and we can actually explore it, right? So we can sample it, as I mentioned, just grabbing random points, or we can optimize. So we can say, what is the point in this space that has the highest growth rate, right? So that for example, is this optimal solution here, depending on, you can actually maximize in any direction you want. But let's say V3 is growth, right? Say this is our biomass reaction. We can optimize in this direction, end up at this point, and this V state is the maximum growth state. This V value is the growth rate, right? So now we've converted our network into a matrix, and our matrix into a calculation of steady state flux values. And so all of constraint-based modeling essentially comes down to defining this space, right? So we define our network through reconstruction, and we define our constraints through mapping data on there, and maybe some other tricks. And we try to predict a point that is as realistic as possible, right? So I mentioned already that we end up with a realistic growth rate when we're just maximizing growth aerobically. That's all we're trying to do, is do this under different conditions, do this with, you know, adding expression data and map that in, and other things like that, right? So we're just, so this is all that there is to it. We take a mass balance at steady state, and then solve it with what's called linear programming by identifying an objective and maximizing it to find this point. So how do we choose our objective? Well, we talked a lot about objectives already. Growth is extremely important to bacteria, maybe less so to human cells. And so in human cells, we often deal with non-growth objectives. We can pick ATP production, for example, is used a lot. Liver cells have to produce a lot of ATP. So that's a fairly realistic growth state, but they also do a lot of other metabolic things, right? That's not energy production. So if you just maximize energy production, you're probably going to get a lot of your network that's not turned on. So your choice of objective is one of the biggest issues and challenges in constraint-based modeling in general, over the history of the field. For example, in E. coli, the models don't predict any DPH production that well because we just don't know that much about what any DPH is doing outside of a couple biosynthetic reactions. So there's still some holes here and a lot of decisions that go into it. Fortunately for biotechnology, it's very straightforward what we're interested in. We're interested in growth because we want our cells to grow under different media conditions. We want to see if our carbon balance is matching very tightly, and we're interested in production, right? So those are the ones that we're going to be talking about most often. In the broader context, you often have to consider other objectives, and we've even talked about methods that don't involve an objective at all, like sampling and flux variability analysis, that just explore this space. So sampling finds random points within this cone. Flux variability analysis finds a cube, calculates a cube around this cone because it maximizes, minimizes in each direction separately. So it's like max V3 is here, min V3 is here, right? So you get, one part of the cube there, so on and so forth. So that's the way these things function. The other piece is the constraints. So how do we get our constraints? I alluded to it already. The biggest source of our constraints for this problem is exchange reactions. If we have exchange data, glucose secretion rates, oxygen uptake rates, CO2 production rates, then we're in really good shape for doing flux balance analysis because when cells are growing fast, they're inherently going to grow near their maximum growth rate, and the model predictions are going to end up fairly accurate. When we don't have exchange data, it's much more challenging. For example, modeling inside of a human tissue, you very often don't have uptakes of all the metabolites that you wish you did, and that's a big problem for predicting any kind of internal state. The other thing that we can choose, so we talked about maximizing our biomass is the objective. But we can even constrain the biomass at a measured value. If you measure growth and you maybe want to see, okay, what other carbon can this be producing? Like what are the possible bioproducts that this cell could be producing that we're not measuring? You can constrain the biomass at a measured value. Another piece I haven't really talked about is there are ATP maintenance costs. There's a special reaction called ATP maintenance that is just the consumption of ATP that I'll talk about a little bit later. That's phenomenological. We observe that these processes are ongoing, and we need to account for them in order to match the predictions. But we don't have a strict definition of what is the source of those costs. And then the last piece is, in very rare cases, we can get internal constraints. So most often, we're not going to be able to measure intracellular reactions. It's very difficult. But there is a data type that can do it, and that's if you label your carbon and do carbon tracing. You can actually say, okay, there's 60% of my glucose is going down glycolysis, 40% is going down pentose-phosphate pathway. And you can constrain that ratio within your model. Or sometimes you can even constrain an exact value. So if you have C13 data, you can do some internal constraints, but most often we're not operating with those constraints. Just one more word on where the ATP maintenance costs come from. So biochemically, it's not a figment of our imagination. It's any ATP that is not associated with growth. So it's not ribosome producing protein and consuming ATP to do that, but is an ATP cost that's not in the metabolic model. So this could be things like the sodium-potassium pump, maintaining ion gradients, or some kind of recycling type machinery function. These are energetically expensive, and we don't have any idea how much of that is happening. So it's not in the metabolic model. But we can set a dummy reaction that is just ATP consumption at a fixed rate that allows the model to, it's essentially a scaling factor. It calibrates the model so that it predicts good, accurate growth rates. And so we have two kinds of maintenance. We have the growth associated, that's this transcription translation, and then the non-growth associated maintenance, which is things like maintaining ion gradients. And how do we actually calculate these? How do we parameterize these? What are these values? Well, we can look at our yield versus the uptake, and we can get a curve like this. And from this curve, we can actually calculate these two maintenance costs. So whatever ATP cost is present when you're not growing has to be a non-growth associated maintenance by definition, right? But the slope of this, how much the costs are affected by your growth rate, should be your growth associated maintenance. So we've done this type of calculation in the past. With every reconstruction, these numbers change a little bit as we get, new physiological data. So I don't know if we have, perfect numbers still, but we measure these values from physiological data and we constrain them. And so, yeah, so this is just a nice schematic of what it means to optimize biomass at the end of the day. You're taking in a substrate like glucose, and then that, in E. coli just grows on minimal media, right? So takes on some ammonia from somewhere else, but this is all the carbon it takes. So as you use this carbon to produce all of the precursors, nucleotide triphosphates, amino acids, lipids, that go into biomass. We define the biomass objective function we already talked about based on the biomass composition of that cell that we measure, and we construct a stoichiometrically accurate reaction where that has units of millimole per gram dry weight. And when we maximize that, it's effectively a single reaction that lumps together all of the precursors and operates at a rate that is equivalent to a growth rate. So it's a really convenient modeling defined reaction. Okay, so that's the core of the theory behind flux balance analysis. It's these constraint-based formalism. You maximize an objective and it finds the flux state that enables you to do that. There can be alternative routes. I've already mentioned that. For example, if there's a parallel path here, the model wouldn't know. And flux variability analysis would tell you that you can have one or the other. So you need those kinds of alternative tools. So is that it? Is that all there is to flux balance analysis? No, there's a ton of ways that you can explore this space with different tools. Just to highlight a couple of these, here's a graphic of sampling where you're just randomly grabbing points within your, these are steady state points within your space with no objective at all. Here's just the basic FBA where you optimize something. This is an example of the pathway analysis. These min spans I was talking about, find these kinds of basis vectors. It's not exactly this, but it's very similar. When you do a gene deletion, you could think of it graphically as removing one of these edges and constraining your space, and sometimes you remove the wrong edge and your growth rate goes to 0, and that's a central gene and you knocked it out. We talked about these kinds of phenotypic phase planes. What else is helpful here? Yeah, flux variability analysis finds this cube around your solution space. And oh yeah, here's one that we'll talk about shortly as the focus of the second exercise, which is identifying knockouts that actually couple growth to production. So you have metabolite production and growth, and as you grow faster, you actually produce more of your compound, which is very unintuitive. So there are various ways of exploring your constraint space. They all tell you a little bit different things about your solution space, but it largely comes down to feasibility. So how much can you do of something, or can you do something in a certain condition or lacking genes? And then uncertainty, characterizing your uncertainty. Yeah. But beyond this, there have been a whole spectrum of algorithms that use constraint-based modeling for a variety of tasks. The simplest are FBA type methods, of which there is no shortage. These different methods often claim to be more accurate. They give better predictions away from maximum growth. There's all kinds of different ways that this basic FBA formalism can sometimes underperform. And there's different solutions people have tried to improve that performance. There are different methods for model refinement. For example, when we saw gaps in a pathway, like a pathway was blocked, there's methods. Oh, yeah, that's gap filling here. Yeah, model refinement and gap filling may be related. But gap filling, you can have a constraint-based algorithm that automatically fills in missing reactions or guesses what they could be. I talked about how the objective function can be challenging to identify in mammalian cells. And so there's algorithms that try to infer what the metabolic objective is based on expression data. So if you look at what metabolic genes it's expressing, maybe that is what it's trying to do metabolically, and you could build an objective function based on the observed expression data. So that's something that our group has done in the past, and I believe others. And then there's various methods, as I've already alluded to constraining a model based on omics data like gene expression data. So building cell type specific models or trying to predict flux more accurately by using gene expression data, differential gene expression data. So no shortage of methods out there to do various tasks. This is a very robust field by this point, and kind of everything has been done. At least as far as the basics are concerned. There's still more to be done with parameterization of these methods. That's a very hot area, proteome allocation. There are still areas that are growing, kind of new branches that are expanding. But that's just a snapshot of the types of things you can do. Now, just to close this off, I want to get into a couple of the specific predictions that are kind of success cases, and then culminating in the growth coupling suite that's going to be the focus of the second exercise. Okay, so now we're talking about this was how do we explore the space of solutions, and now this next section is where have these predictions been successful and what kind of predictions can they make? Okay, so one of the first studies back around 2000 in constraint-based modeling, metabolic modeling, tried to predict acetate overflow based on at evolved endpoints. So here we have an oxygen uptake rate, and you could see here that the model accurately predicted how the cell would produce more and more of this so as it grew faster and faster and consumed more and more oxygen, it would produce more and more, take up more of this and produce more of a substrate. So, of a byproduct. Sorry, misspeaking. But this is one of the earliest success cases that showed two key things. One, metabolic models can make accurate predictions quantitatively, right? But two, it showed a key feature of these models, and that's that growing fast is a key feature of their success. That because you're optimizing growth, this is like the fastest it can possibly grow, your cell really, your experimental in vivo cell really has to be growing fast for the model to be predicting accurately. If your cell is in lag phase, if it's stressed by pH or something, these baseline models are not going to predict the state or the growth rate that particularly well. This is an evolved strain that was evolved to grow faster and faster, and as it did, the model got more and more accurate, which is the expectation because the model is supposed to be predicting max growth. So this is a very classical paper, a very classical result. Another thing that they can do is not just predict secretion, but they can predict uptake, and they can predict what your cell can grow on, right? So for E. coli K12, this is all kind of well established. People know that like it has trouble growing on citrate for some reason. It's a very weird phenomenon, but like these things have literature understanding. But for other strains, other organisms, it's not clear what this strain can grow on beyond like glucose, there's all kinds of other alternate carbon sources, nitrogen sources, etc. And that's one of the main differentiators between different strains of a species and across species is what they can actually grow on. So this is a study that we did some years ago, building metabolic models for different strains of E. coli and Shigella, which is related, and then calculating whether or not they can grow on different substrates. So these are different growth supporting nutrients, and yes, no, whether or not the strain can grow on them. And if you cluster this, what's interesting is that not only are there obvious clusters, but the clusters align with the whether or not the strain, the origin of the strain, whether or not it's pathogenic. So you have intestinal pathogens, commensal strains, meaning non-pathogenic, and then extraintestinal pathogens. And so I alluded to this earlier with the tagatose study, but whether or not a strain can grow on a particular substrate is indicative of the types of environments it can thrive on, right? Because intestinal versus extraintestinal are going to have very different nutrients there, because different microbiome neighbors that are sharing different metabolites, different diet, the kinds of material that is there to survive on is different between these environments. And you actually see that reflected in the metabolic content of the organisms, of the strains that like to grow in these environments. And who knows what the implications of this are. There could be implications on antibiotic resistance, on virulence. There's a lot of possible angles here. And so with this type of understanding, you can even build a simple decision tree that, based on just a couple key metabolites, predicts whether it predicts the location of that a particular E. coli strain will settle in. So you can think of these as key nutrients that might be like you would assume that these would actually be differential in these environments themselves, or related metabolites. And that's actually why these organisms have chosen to keep these particular metabolites. So another very interesting type of prediction that these models can make is what the cells can grow on, and has some serious clinical implications. Probably the prediction that these models are best known for is predicting essentiality following a single gene or double gene knockout. And you can see here the accuracy of single and double gene knockouts as the E. coli model has progressed, and also for different other models. And you can see that the accuracy and number of predictions that it's made has increased over time gradually. So the model can predict whether or not a gene is essential on particular substrates. The double gene knockout essentiality is actually not as good to predict, which could be interesting. It could be route for discovery, or it could be indicative of experimental issues with double knockouts. Could just cause more regulatory issues. But this is one of the very common predictions. There's also, just been a plethora of different methods trying to use metabolic models in humans, either to integrate gene expression data to build tissue-specific models, to build models, that map between human and mouse. So there's a mouse model that's been derived from the human, to take gene expression data and try to understand how drugs or disease are perturbing a cell. So if you induce a cell with a drug, it's going to change the gene expression. You can predict whether or not that's going to affect the metabolic state, or which pathways might be affected by that. We predicted things like nutrient deficiencies due to drug treatments. That's one of the predictions that these models have made. And you can actually simulate a drug effect by simulating a knockout. So antibiotics, there are metabolic antibiotics that target metabolic genes, and you could predict essentiality. There's also just genes that affect metabolism in other ways that you can simulate with this. So there's all kinds of predictions that you can make. Maybe it's not worth going into all of this right now. But this brings me to the last prediction, which is also the subject of the second exercise for the day, which is growth coupling. So growth coupling is a very interesting phenomenon. Normally, you would expect any two processes, metabolic processes within a cell, to compete with each other if they're both production. For example, you could produce acetate or you could produce lactate, but you can't produce a maximum amount of both, right? There should be a negative relationship between production of the two. So growth is the same. Normally, the more you grow, the less carbon you have available to produce other compounds. So normally growth competes with target production. Now, this is a huge issue in biotech because we often, we adaptively evolve our cells. So they grow faster and faster. And as they grow faster and faster, they produce less and less of your pathway because they've evolved, they've self-selected to grow fast, and that would inherently down regulate any kind of heterologous production. And so that brings me to growth coupled production, which is for particular designs through knockouts and sometimes knock-ins. You can actually set your network up in a way so that Flux has to produce your target compound in order to grow. So you can actually couple your growth to the production of a compound. And when you do that, then, in order to grow faster, your cell has to produce more of the target compound, right? Now that is great because not only is your cell robust, so as it grows faster, it won't stop producing, but you can even select it to grow faster, or sorry, you can select it to produce more just by setting up this design and evolving it in a lab to grow faster and faster, right? Because it'll self-select to grow fast, and by growing fast, it'll produce more of your target compound. That's way more efficient than screening, than generating a bunch of variants in screening and then more variants in screening and directed evolution, that type of thing, right? This is just self-selected. It'll figure out whatever mutations it needs to grow fast and produce this thing. That's great. So growth coupling, very exciting concept. At the end of the day, you still are using a bunch of your carbon for growth, but for some compounds, that doesn't matter, right? It's just a matter of getting activity. And we'll also discuss a case of enzyme selection where the target isn't even production of a compound. You're trying to just put a selection pressure, a growth pressure on an enzyme. And there you don't care about this, this trade-off at all. And so so what we're looking at here is a production envelope. And let me describe a little bit of what this is. So on the y-axis, we have production. On the x-axis, we have growth rate. Now, the shape here is the constraint space. So everything within this is a feasible growth rate, right? But it's possible. So normally you see this trade-off. As you grow faster, the amount you can produce goes down and down and down, right? So as you grow maximally, it produces 0. So that's terrible, right? You've grown a really fast strain, but it doesn't produce anything. So that's kind of useless for your biotechnology application. But with certain knockouts, you can reshape your space. So we talked about how knockouts restrict your constraint space. So it X's out, it removes this gray space here. In your new space is this only the blue, right? So at 0 growth rate, you can produce no compound or a bunch of compound you don't know. But as you grow faster, the design has this interesting phenomenon. And I'll show you an example of how this can work metabolically, like what the biochemical logic is behind this. As you grow faster, in order to keep growing, your cell has to produce the compound. The minimum value here increases. So it can produce a bunch still, but it has, it at least has to produce a little bit. So at the end of the day, you'll end up with a strain around here. And then that is a fast growing strain, relatively fast, and still produces your compound. So this is the growth coupling. As you grow faster, it has to produce more. And there's different kinds of growth coupling. So there's weak coupling that it only shows up after a while. And sometimes it's so weak, it's like right at the end that it's really hard to actually ensure if that's actually real. There's also this holistic, meaning it starts there, and like strong, which is high production even at low growth rates, so there's not really a growth selection there, but it already produces a bunch, so I guess you don't care. The point of growth coupling algorithms is to identify knockouts that actually create these situations. So there can be combinations of genes that create this kind of shape, but we don't know ahead of time. So we have to have some kind of algorithm. One of the earliest algorithms to do this was OpNock. This is published in 2003, I think. OpNock has a more complex formalism than just simple FBA, but at its heart, it is just using SV constraints It's just a bi-level optimization they have to solve with a mixed linear program. So it's kind of a pain to solve for large deletion sets. But anyways, it is at its heart a form of FBA at the end of the day, but it selects knockouts that reshape your space so that you're producing your compound. And so this is just one example here, a lactate producing strain. So you can see they knocked out the phosphotransferase system and some of these alternative routes for dumping redox equivalents. And so by doing this, the only way it could get rid of the redox is by dumping them on lactate. So to regenerate NADH, it has to do it through pyruvate and lactate. And so in order to grow, it has to regenerate NADH. In order to regenerate NADH, it has to do it through this reaction. And so it just produces a bunch of lactate in order to grow. And so the effect of this on the production envelope, you can see here. So you have the wild type here that has this behavior that I mentioned, that at high growth, it produces nothing, right? So that's terrible. But then these designs can create this kind of situation here, where as you grow, you have to produce a bunch of lactate to grow, right? So as you keep making these mutants, you end up with this really nice strain that as you grow it, it only grows at 0.1, so not super fast growing, but fast enough to double and acquire mutations and run an ale on. As you grow it, it'll produce a massive amount of lactate as a stable strain. So this is how this concept works. OpNock is one way of doing it. The CFB also did this procedure. I think this was designed manually, but this is same type of thing. The production envelope for a methyltransferase system that the CFB designed in order to build an enzyme selection system around the SAM cycle here in E. coli. You can see here, this kind of has that feature where it's kind of weak coupling. It only shows up at extremely high growth rates, and until then, you're not going to get very much selection for higher production. So this is kind of not a very desirable phenotype, and it really has a high demand. So, so even though this was manually designed and it worked, maybe it has some features that we'd like to improve by computational prediction. And so that's what Tobias's work is, and that's what he'll describe in his workflow. So I'm just giving a quick prelude here. So his method is kind of an improvement on manual methods, and the goal of this method is to find designs that maximize the minimum production rate at a set growth rate. So you set growth rate as some tolerable level, say like 0.3 or something, where you can evolve this and it's not going to be so low it causes problems. And then you maximize the minimum value to get this strong coupling situation so that you know that it produces your compound. And you can do this for a variety of purposes. I don't know which ones he's going to go over today, if he goes over production and enzyme selection. But you can do this for, you can evolve a variety of metabolic phenotypes with this procedure. And so I'll let him take that away from this point and end this second lecture just summarizing what we can do with metabolic models. So in the first part, we talked about metabolic network reconstructions. That's the network, it's how you get it, it's the knowledge, it's how do you believe in it, and how do you make it as accurate as possible. In this section, we talked about how do you make predictions with that? How do you convert it into a mathematical model and simulate with it? We already went through CobraPy and Escher FBA and as examples of that, and hopefully you feel comfortable utilizing these models moving forward. But the key steps are you take your network and you convert it into a model by, one, constructing your stoichiometric matrix, right? That matrix S is critical to basically every algorithm. So the nice thing is it's not difficult to construct, it's just directly the reactions, respecting reversibility and elemental and mass balancing. And then two, you have to define your constraints. And so reversibility I already mentioned, but also any growth constraints, any exchange constraints, any kind of data you can use to constrain your model to keep this, because the S matrix by itself is this unbounded problem. And then you identify an objective. Fortunately for E. coli and for microbial biotech, we're either interested in growth or production most of the time. In mammalian cells, it's much harder. So that's not a can of worms I want to go into too much here. And then there's a variety of predictions you can make with this. We talked about predicting overflow metabolism. We talked about predicting growth, no growth on knockouts, growth capability on different substrates. You can do a variety of predictions with just that. And there's been all these different tools built to explore these constraint-based spaces. So Flux variability analysis, sampling, knockout prediction, different methods for inferring objectives. There's a slew of them. And then just last, these growth coupling predictions are one of the more interesting algorithms for design, because you're building strains that are robust experimentally and not just a figment of your in silico imagination. It's that you can do these knockouts and build that strain. It's guaranteed to grow at a high rate and produce this compound stably, as long as there's not some kind of leak or some kind of something that the model doesn't know about. Okay, so that's it for this section. I will take any questions, but if there's not any questions, I'll pass it over to Tobias to go through his growth coupling workflows as a more advanced application of CobraPy.
Awesome. Thanks, Dan. Also, thanks for the pretty extensive introduction into the metabolic models, into reconstructions, and also the growth coupling algorithm that I'll be showing some hands-on in the next couple of minutes. I haven't heard from anybody about the growth coupling suite, or if there were any other problems. So I assume it is all right, and at least the modules can be loaded. I would just go through the different parts of the first example, the basic example, as I call it, and I think afterwards we let you some time, give you some time to do it yourself, or if you have it in parallel, you just click through, and after I finished, you can ask any question. You can ask any question anyways at any time. But yeah, I think after I finished, we just give you some time, give you some hands-on, try different things out. Maybe you want to change something in the script, try out different stuff, and yeah, we troubleshoot if you have any questions, errors, or whatever. Yes. So the first comment about the growth coupling algorithm. This is somehow ongoing work. So the algorithm itself is published, but the modules and the Python implementation is not as of now, which is not a problem. I will show it you anyways here. So it basically consists of two things, which is, well, I adopted GCOpt. This is the growth coupling algorithm that takes the M model and basically gives you a design, or optimizes a design for growth coupling. And this is not really related to the growth coupling algorithm, but kind of a strain analysis routine where the output from this GCOpt module is taken, analyzed, and somehow put into context. What kind of designs were found, what interventions are within these or make up these kind of designs, and what are the parameters, maximum growth rates, something like growth coupling strength, and so on. I will show this to you later. Yeah, so get right to it. First thing that we do normally in a script, and Dan has already shown you with the CobraPy examples, you load the modules and package here. I just go through it. If you don't know, I have any experience, I just activate or run each cell by clicking on it, activating it, and then Control-Enter, executing it. So lucky me, there's no error. So obviously the growth coupling algorithm and this strain design tool and other modules, Escher, and of course CobraPy has been loaded. So what we first do, and this is a basic routine for metabolic modeling in general. If you use growth coupling algorithms or any other thing, OpNot, you name it, we of course load a model and set up the model, prepare the model, set the constraints as we like. So here I simply define the pathway to find the models. You see the structure that I have. This is the script that we just run and see right here. And within models, well, I have put some of the models that we're using. And today we are actually using a core model of the latest E. coli K12 reconstruction. So core means that it has been stripped of some redundant reactions for the most basic growth states. So this model actually only has I think 600 reactions left from the 2,700 from the original IML 1550 model that Dan has introduced you as well. So what we do, we load the model and we set some constraints. So we basically set the environment of the cell that we live in. That the cell will live in. So we do that. So now it's loaded, and these kind of constraints has been set. So just to explain a little bit further, so this model that we just loaded have these reaction classes in it, and by this function get by ID, you can search for reaction that have a particular name. Here, for example, the exchange reaction for the glucose, and we set this to a lower bind of -18, meaning that the cell can take up glucose with a flux rate of 18 max. We also set here the oxygen uptake rate, and this is particularly important because it's this oxygen uptake rate, or the limit of this oxygen uptake, can drive a lot of things. Normally is set to -20, which is about the let's say physiological or thermodynamically possible uptake rate from air to the medium and then to the cell. I set it here actually to -2 because we work in anaerobic or kind of microaerobic conditions. And why we do that, I explain in a second. And this one is just to avoid any peculiar behavior of the model. I don't allow for any any uptake of CO2. So what we are going.
Can I ask you a question?
Yes, please.
Is it quite common to remove the CO2 uptake in the metabolic modeling, or is it, why did you specifically
yeah, it happens that in some examples, when you design strains, when you analyze the flux by flux balance analysis, you see that CO2 is taken up, which is, I believe, quite unlikely. I mean, we've seen efforts where the uptake of CO2 is somehow enabled, and E. coli can grow actually on CO2, but I would like to avoid this. But this is really in rare cases, so you don't necessarily need that. It depends. You probably don't have to do this. If you see that your design strain takes up CO2, then you have to ask the question, can that actually be? Yeah, or is this just because there are some reactions in the model that can integrate CO2
kind of
within the carbon
reaction to synthesize oxaloacetators.
For example, yes. For example, this anaplerotic reactions. And I think for most cases, it's highly unlikely that the cell takes up CO2, net CO2.
Okay.
Yeah. So because what we, and this is the example that we're dealing now, what we want to do is we want to couple 1,4-Butanediol to growth of E. coli. So 1,4-Butanediol is kind of a commodity chemical, which can be used for polymers, for special polymers synthesis. It has quite a market, and I think the production, the microbial production of 1,4-butanediol is also established on an industrial strain, on an industrial scale. And all the data and information about synthesis pathway and anything about this 1,4-butanediol is from this publication right here. We can have a look. Probably you know it. It's about 10 years old right now, I guess. So what they did, they went through the effort of constructing a pathway for the 1,4-butanediol synthesis in E. coli, because E. coli cannot do it natively. And engineered the cell with knockouts and so on to overproduce 1,4-butanediol, grown on, I guess also here it's glucose, I believe, glucose xylose as well. So we use this example, and this, what you see here, is the synthesis pathway that has been identified by this paper. So here you see these. This one is actually a E. coli native reaction, and all the numbers here you see in red, I believe, are heterologous reactions from other organisms that were or has to be integrated into E. coli to enable the butanediol synthesis. So the first step for us is, of course, to model this kind of synthesis pathway in E. coli, or to integrate this pathway into our model. So the first step, and Dan has already shown you how to integrate reaction in general. The first step is to introduce this kind of this reaction step here. You see it here. I don't go into detail. So this is succinyl-CoA to succinyl semialdehyde. And there are actually two steps that can be used to produce this succinyl semialdehyde. So the primal precursor of butanediol. So the second route is from alpha ketoglutarate. You see this reaction here. So we construct the reaction, we add the appropriate metabolites with the corresponding stoichiometrics. And this is done later all together, I believe. Yes, and we add all the reactions later to the model at once. One thing that I did here is I did not specifically model each reaction step here, but I lumped this whole pathway into one reaction. Why did I do that, or why can we do this? This is because these intermediates, succinyl semialdehyde, that's not true. That's actually already here, but these precursors here are not native in E. coli. So at least the model doesn't know what to do with it. So there's no need to specify these particular metabolites. So there's also no need to introduce every single reaction. But what we can do a net balance of all the cofactors that I used and produced. Here's acetate produced and acetyl-CoA. Here's another step that uses NADPH or NADH, releases CoA again. Here's another drain for for redox equivalence until we actually reach butanediol. So this is all squeezed into this one reaction that you see here, and you can actually see it via the relatively high stoichiometric factors for NADH, and I actually used only NADH, not NADPH. So you can already see by modeling it that the synthesis pathway actually has very high redox demand. That's just let's say a side fact. What we also normally need if we introduce a non-native substrate, a substrate that is cannot already be found in the model, we need to define an exchange reaction. And actually, good practice is also to define a transporter for this kind of substrate. Well, in this case, I did not went through the hassle to identify actual transporters that can do the job and identify possible mechanisms by which butanediol is transported via the membrane. I just assume here that intracellular, so butanediol in the cytosol can simply be exported over, well, in this manner, but in this example, over 2 membranes into the extracellular space. So we don't assume any proton pumps or maybe even active transports. But if you want to be super precise, you should definitely do this, because I think Dan has mentioned this at one point. A redox or even a proton balance can be very important for models as well. So we create all these reactions, this exchange reaction, which then shuffles or transports the butanediol out of your system, some from the extracellular medium, so somewhere into the medium. We integrate it all, and now we're able to simulate some basic properties of the butanediol synthesis pathway. I think I didn't execute this already. So all right, so this is done quite successfully. Great. Yeah, so what we do now, oh, I already did that. I will simply execute it here. With basic properties, I basically want to simulate the flux-based projection. So the projection of butanediol production rate with different growth rates. And I actually have a method implemented in this module here. So this is the strain design analyzer, the custom-made strain design analyzer that comes with the growth coupling suite. So this is loaded here, strain design analyzer. I call this here SDA. So I basically load this analyzer with the model that we've just prepared with the heterologous pathway, the butanediol synthesis pathway, and yeah, we load this analyzer with this model, as I said, and that what we can do here, there's a method within this analyzer that says flux-based projection, and you can specify the target reaction. So the target reaction would be the butanediol exchange reaction or the production reactions. And we can specify, I think I didn't do this here explicitly, but the model assumes that that on the x-axis it has to plot the growth rate or the biomass equation. So what it simply does, it calculates the space, and what we can see here, that given the constraints that we set, meaning a glucose uptake rate of about -18 and a rather low maximum oxygen uptake rate of -2 millimole per gram cell dry weight per hour, we can reach a growth rate, a maximum growth rate of 0.4 roughly at 0 growth. So the maximum production rate of butanediol is something about probably 18 millimoles per gram cell dry weight, which translates to a yield on glucose of about 0.5 gram butanediol per gram cell dry weight. So this is nice to know, okay, what is the cell capable of? Can it actually do the job? And you see here for different growth rates, it's of course possible to have an active butanediol production, and quite a significant one as well. But you also see, and which relates to the growth coupling topic that we're dealing now, you see that for any growth rate, this lower bound here is straight 0. So we cannot expect that the Butanediol production or synthesis is somehow coupled to growth, or will occur if we, for example, allow strains maximize the growth performance of the strain. So, okay, before going into detail about the growth coupling, here is, and Dan promised this also, here is, all right. Here is the Escher map for the let's say wild type model in terms of the model with the integrated Butanediol synthesis pathway, and a flux distribution where I maximized the objective, meaning where I maximized growth rate, and I actually used PFBA, so parsimonious flux balance analysis. This particular method maximizes growth, sets this objective value, so the maximum growth rate, fixes the maximum growth rate, and then minimizes all the other reactions. This is kind of a resource allocation problem that is solved here, because the assumption is that when a cell grows, it uses as less enzyme as possible, and that's done by minimizing the sum of all fluxes in this case. So here, and this is very great about the Escher map, here you see an overview, and I will quickly zoom into, get into full screen mode. Obviously you don't see much. Hopefully it's now better. So what you see is glucose is taken up via a PTS system. Actually here it's not using pentose phosphate pathway at all. It's debatable if this is really making sense here. The models are not, or metabolic models have in general a problem to really get a physiological relevant distribution between glycolysis flux and pentose phosphate pathway. Another complication is that we're using a truncated model, so this is just to exemplarity reason. Just an example here. If you really want to know what's going on, use the full model, but all the computations would take much longer, at least when we use the growth coupling algorithm. That's why I chose also a simpler, smaller model. So you see glycolysis is used here. And what you can also see here is that's kind of interesting, because it's this one also uses the TCA cycle somehow in a backwards fashion, or this looks, at least to me, a bit funny right now. So yes, the main reason why this looks funny is, of course, because we're using kind of microaerobic setting, and these kind of models are not super adapted for this particular phenotype or environmental condition. But at least you get a feeling what the model thinks is, for example, being secreted. So you see a good part of carbon is drained via the acetate, via acetate drainage. Also format is produced, and ethanol. So you get a good sense that that the cell is here struggling actually for ATP. So okay, so Dan was introducing actually the growth coupling principle, and very briefly. So I will just explain it a little bit more on this figure here. So you know the flux spaces already, production rate, in this case Butanediol, and growth rate. And what we want to achieve, we want to find a strain design based on knockouts, and also heterologous insertion of heterologous reactions. We want to find a strain design that raises this lower flux bound here to a level where where for any kind of growth rate or growth state, the cell has to produce our product. So it has to produce Butanediol. As you can probably imagine, there are a lot of opportunities and several strain designs are possible, but we want to find kind of an optimal solution to that. So what we do, or what the algorithm does, it fixes the growth rate at a certain point, and then it tries, for this particular growth rate, tries to find the maximum production rate for Butanediol. So first it minimizes actually the Butanediol synthesis rate and then tries to maximize the minimal Butanediol production rate, which translates to it tries to maximize this lower flux bound for the Butanediol production rate here. Let me just zoom out a little bit. So, and how are we doing this? How is the setup of the algorithm working? So all this, let me just add to this. The nice thing about this principle is also in comparison to, for example, OpNoc, we don't assume a specific cellular objective here. So it means we don't assume that the cell has to grow maximally fast, but the objective is kind of network based. So we really want to find a solution where by stoichiometry, it's not possible for a cell anymore to get around product synthesis when it's growing. So with OpNoc, then explained to us real shortly, it relies on also, for example, maximization of growth. So it tries to find a solution at maximum growth, which could be a bit biased, because when you, for example, introduce deletions, maybe the cell won't grow maximally fast again. Maybe after ALE, of course, again, but here you're doing absolutely unbiased optimization of your design in terms of growth coupling. So what are we doing to set up the growth coupling algorithm and get this running? So we have set up the model and all the constraints. So we set up the environment the model will live in. And now we have a config file that we fill with a couple of parameters to run the algorithm. And I will quickly here, this config file is imported. You see here the config basic.py file, and I will open this here. Here, just some general setups. We can set the output directory for our results that the algorithm is hopefully producing. We can set an ID of the results file. This is quite important. We have to specify the ID of the biomass reaction. Normally, of course, we can assume that the current objective in the model is the biomass equation, but by explicitly specifying this one here, we make it explicit to use particularly this reaction ID as the biomass equation. And this is also important. I just told you that the algorithm will fix the growth rate at a specific rate. And this you can actually define here. So this is the growth rate in per hour. In per hour. You can set this here. So this is quite low. I would also recommend it to be rather low to find really somehow strong designs here. You can set a time limit. So our simulations or optimization will run for about here 300 seconds or 5 minutes. I will just set this maybe to 200, that it runs a bit faster. And also to not overload my computer or laptop here, I only use 3 processes. So the whole optimization runs on the Gurobi solver here, which you hopefully managed to install. And this one can actually use all your processes of your machine to do these calculations and calculate optimal designs here. Just talking about a laptop, just a tip when you when you work on the computer and do modeling stuff, always save your stuff that you're working at runtime at best, because I had to figure out just before the weekend that your computer can crash quite ultimatively and quite fast. So this just a good tip for you. This one is also a very the total intervention. So the number of total interventions is a critical parameter, because it defines the complexity of your problem. I set this here to 5. So in total, 5 interventions are allowed, meaning in this case, 5 deletions or addition of reactions. And you can actually specify within the realm of 5 interventions what are the maximum numbers for deletion and add-ins. We come to these ones later. So I set basically setting both parameters, so both the number of deletions, the maximum number, and the number of add-ins to 5 means that the solver or the growth coupling algorithm itself can choose whether it uses deletions or more add-ins to arrive at an optimal strain design here. I don't go, since this is just a basic example, I don't go into details with these kind of parameters here. You could also well delete it here. So this will lead to the algorithm taking some default values. What you can do here, this may be somehow interesting. You can exclude subsystems. So all the reactions are clustered into subsystems in the model, and you can say, hey, this subsystems, well, exchange reaction or cell envelope biosynthesis. So all the reactions belonging to cell envelope biosynthesis should not be targets, for example, for deletions. This is also not that important right now. Let me just go through it. Yeah, this one I would also not touch, but just to make you aware of it, we improve, or the algorithm before starting the optimization improves the numerics in a way that it adapts the biomass equation a bit. So in the biomass equation, there are metabolites which have very low stoichiometric coefficients, or the range of stoichiometric coefficients is quite broad. And mathematical solver doesn't like to deal with large and low numbers at the same time. So I erased some of the insignificant metabolites in this biomass equation to arrive at more plausible solutions. Because it can happen if you don't do this, the solver will find solutions that are actually infeasible for the model if you test it otherwise. But this has numerical reasons, let's say. So yeah, so we went through our configurations file. So just to recap, we said where we want to store results, and we said how many deletions and add-ins we want to consider in our strain designs. So this is the basic. So we load this kind of configuration file, and I will start this right away. What we do here is we load the growth coupling algorithm class with our model, and actually use a copy of the model with the Butanediol synthesis pathway. we need to specify the target reaction. So this is the target reaction that has to be coupled to growth, and I obviously specified this. Yes, exactly here. So this is the exchange reaction for Butanediol, which we ourselves defined here and set up here and integrated into the model. So where were we? Sorry, here. Yes, and then we pass also the configurations file that we just had a look at. And we said you don't necessarily need to specify this, but we want to set up the GC op problem. What's happening now inside? So the algorithm is actually still running. So what's happening? The model is taken, and it's firstly the model or the solution space is reduced a bit. So given the environmental conditions and the constraints that we set, essential reactions for the conditions that we've chosen are actually removed from the target space. So these cannot be knocked out because we know and tested beforehand that we need these kind of reactions. Very trivial example is, for example, the glucose update. So we need the glucose transporter, for example, the PTS system. Okay, we don't necessarily need it because we also have the ABC transporter, but let's say the glucose exchange reaction, we need this kind of reaction, so there's no need to knock out. There's actually also another reason, because there's no gene encoding for an exchange reaction. But for example, we need the transporters, so we need to leave them in the system, and there's no need for the solver to test if it's a good idea to knock out, for example, a transporter for glucose. What we also did, because here in the config file we specified that we also want to test the integration of heterologous reactions. So what it's done internally, and you see it somehow cryptically, sorry, this is a bit, you see it, where's it exactly? Here. So this says that a database for heterologous reactions for this particular model that we use is being loaded. So what I did, and I did this before because this takes considerably amount of time, I scan all the reactions from the models that are in the big database. I, let's say again, look for the thermodynamics of the reaction. So I curate the direction that the flux for this reaction can take to be very sure that the reaction that is annotated in the original model is actually also a reaction that can happen in an E. coli setting. So internally, there's also concentrations for, or most likely concentrations for cofactors are used to arrive at plausible reaction directions. And what's actually done is the equilibrator database is consulted. And maybe just a quick excursus, equilibrator. This is a very nice tool that you can use in Python, but it's obviously also a very nice web page. This is done by the Weizmann Institute, if I'm not mistaken. So here you will find thermodynamic information of probably almost any kind of reaction. So let me, there's already, so you can, for example, search for pyruvate dehydrogenase, and you see several versions for it. I just click on the ones that we probably know. So this is this one here. And what you see here is, and that's what Dan has told you, this is the Gibbs free energy of reactions for this kind of, for this reaction. So for the oxidation of pyruvate. So this number here, so the minus 34 tells you that this reaction is most likely happening from left to right. And what you can do here also in the website, you can enter, right, you can enter custom concentrations for these different metabolites and test when do we get actually positive numbers here. So positive numbers means that actually the reaction would go from right to left, so simply the other directions. And if you test for example, crazily high amounts of products versus low amounts of substrates, and you still have a negative delta G, then you, for example, can assume that this particular reaction is irreversible from left to right. Just check it out. Equilibrator, great tool, and you can actually also use it in directly in Python and use it via an API. So pretty useful. Yes, so where are we? So this is actually done. This is good. Where have we been? Right, we have curated our heterologous reaction database. So we integrate all the heterologous reactions into our base model, and we let the algorithm then decide which of these heterologous reactions to switch on. And it will then find the best combinations of deletions and switch on heterologous reactions that enable this growth coupling in this kind of sense. Okay, so we've set up the GCOF algorithm. Actually, here you can see this is a bit sensitive. You can actually see which kind of metabolites were removed from the biomass equation, and you have the stoichiometric coefficients for that. Just to let you know, here's some stuff and about setting up the optimization algorithm. Yes, and here you have a check about the complexity of the problem that you will face. So you said we want to have a maximum number of interventions of 5 out of 1,160 almost. So we wanted to allow deletion targets between 0 and 5 out of 170 targets. So we have 100 in total, 170 potential deletion targets, and our database has almost 1,000 heterologous reactions that the algorithm can also choose from. Okay, so this class is loaded. The only thing that we now have to do is to optimize this kind of GCOP problem. So you have already seen almost the results here. So if you press start, then it should immediately start running. And if you see something like this is already the first good sign, because this means that actually the solver Gurobi started working. Right, so how many seconds did I put here? 200, all right. So we have some time. These lines are actually filled up one by one after some time. Maybe for the more for the ones that actually have seen something like this before, are more used to models. What you see here is in these columns are the objective bounds. So when you remember, the objective here was to maximize the minimal guaranteed production rate at this fixed growth rate. And the value of, let's see, here we are. And the value of the current best solutions for these objectives you see in this column. So this is the maximum possible, and this is the objective of the currently best solution. You see here in the beginning there's 0, but you see there's a, well, callback solution. That means that the solver has found a solution and we have checked the validity of the solution. Actually you see here that there's a solution which has a guaranteed lower butanediol synthesis of 0.2 millimoles per gram dry weight and hour. So this is already a good sign. So if you're dealing a lot with this kind of algorithms, this is your happy moment, right? Because you know, okay, there is at least some solutions which you can analyze at the end of the day. So this is already great, and you see it's progressing, and it's actually also finding, the algorithm is finding even better solution building on this one. And the nice thing about CobraPy, particularly for the Python implementation, is that it also solves these intermediate solutions here. So you can also have a look at all the solutions afterward, not only the absolutely best one at the end of the simulation. All right. Yeah, I think we have 40 seconds left until this one is finished, and then we start with analyzing the design solution. So what will happen if this algorithm stops running is we have a results folder here, and I specified also in the config basic, I specified, yeah, put our results into results and the Butanediol basic folder. And okay, I see I had previous results here, so I was running the algorithm already before. You have one folder, callback solutions. This is actually the current solutions that came up. And okay, I was just confused about the file IDs. I will tell you in a second why. And here, in fact, when we analyze later all the results, we also we will also get Flux-based plots for all the designs. And at the end of the day, we will also get an Excel file with all the design solutions that the algorithm found on its way to the optimal, or let's say suboptimal solution that it was able to find during the time frame. Just shortly why I was confused for a second, because I actually put here the output file ID to I7, K7, A7. What I normally do is I label the simulations file by the number of allowed total interventions, just to have some overview. So here, obviously, I didn't adapt this to the actually 5 interventions that I put here. Anyways, I think it should be done by now. No, not yet. Okay, are there any questions so far? Is anyone running this script in parallel as well, or are there any comments?
Yes, I just have a, I don't know if we have time to get into it, but I'm curious about, have you looked into how CobraPy stores the objective, or stores the solution, like meta information, like constraint tolerances, whether anything was violated, feasibilities? Because at the end of the day, is this an MILP? Is an LP? What is it? What is
yeah, I didn't go into the details into that. It's an MILP. Just for maybe for some of you, MILP. So what's done here, the way I explained it is that we have 2 linear programs, two objectives that are solved at the same time. So we first we minimize, in this case, Butanediol production rate, and then we maximize this minimal production rate to arrive at higher, lower flux bounds here. So these are 2 linear programs, and there are actually mathematical ways to integrate it into one linear program. So that's done by some techniques. And why integer? Because the deletions and also additions additions of reactions are assigned by integers, or in our case booleans, 0 or 1. So if we have, for example, a 1 for a particular reaction, then this one is, for example, activated if it's a heterologous reactions. Or for the deletion targets, if any reaction is assigned a 0 by a variable, then it's deleted, for example. So this is an MILP, and there are quite a lot of metadata comes with every solution, and this is also a nice thing for in Python, also in comparison to the MATLAB version of Google, because it allows you to do anything with the most current solution. So you see here, it's maybe to make it more clear, here there's the current best solution is actually 0, so it has no solutions. And then something happens. It says evaluate callback solution. So the Gorobo solver says, I have a solution, and then it actually allows you to run any custom code with this solution. So it returns this solution. And this is actually printed based on a function that I myself wrote. So what's actually done here, I take the solution, I take the original model and test this solution, this design solution on a model that is not somehow crammed into an MILP. So this is very great. And I can also immediately assess if this is the solution that has been returned is actually a valid one in a pretty standard metabolic model. Right. And we will see later in the advanced examples that you can go quite crazy with these callbacks of the solver.
Okay, thanks. Yeah, I was just curious because I've run MLPs and seen these kinds of trails, and sometimes I'm speaking from the MATLAB version, it'll throw warnings like, integer tolerance exceeds this, and it just says warning. And I'm just trying to understand, how to interpret those things. I was curious if Python had better tools there, or if you had looked at those, like to make sure these things are not violating constraints and things like that.
Right. This can be sometimes very cryptic if you deal with the MILP in particular, because your model is transformed and the stoichiometrics are sometimes even multiplied, so, and which often leaves you with kind of bad numerics, at least for the MILPs that we're dealing here with the metabolic models. That's also why I did, now you see it actually here, why I, for example, threw out some metabolites from the biomass equation with very low stoichiometric coefficients, because yeah, it sometimes creates problems which you only see after you evaluate a solution on a standard metabolic model. Right. So you can actually do this right away.
Thanks.
Sure. All right. So, but now finally, I think, yep. Okay, completed. That reads awesome. Right. So we see the solver was able to find a solution. And now, of course, we want to know what kind of solution that is, and we want to analyze it. And therefore, we use this strain design analyzer that I've been implemented more or less, particularly for this growth coupling algorithm. So what's done here? We're already in the results folder. Right. So there's right. There is, and you see this is 6 minutes ago. This final results file has been constructed by the GCOpt algorithm. So there's all the information about the found designs in it. And this is what we load with the strain design analyzer. So we direct it to this particular results folder. Exactly, and what it actually does, it reads all the files, also all the files that reads somehow GC-opt solution, and then any cryptic abbreviations here. So it reads all the files because it assumes that any GC-opt results file in this particular folder that has been specified belongs to this one particular problem that has been optimized. So if you want to analyze designs, make sure that the results file only belong to this one problem. So one production pathway or whatever you want to growth couple. Yes, what was I right? So we load this file and then we do a growth coupling summary. So, and I will start it because this is actually can also take a bit. So it will get into this file, take out all these strain designs, and then analyze. Firstly, is this solution valid? It tests again, yes. Are these solution valid? Then it actually also determines if this design is significant. So it tests if some of parts of the design, so some deletions or add-ins, can actually be left out and we still have the same growth coupling result. And if it's the case, then the algorithm immediately throws out this particular deletion or add-in and reduces the complexity of our solution. Well, it saves results. That's also quite nice. And yeah, we come to this later. And it also saves, as I've been saying, the Flux-based plots for each of the design. So here there's a lot of stuff that is printing. There are some errors which you don't need to worry about right now. It's actually still running. So what's done now is that the single designs that has been thrown out by the solver are now analyzed. Wait a bit, it shouldn't be too many. Yeah, it's actually only 3. So you have you see a result here, but you can actually, and you see seconds ago, you can actually observe all the results in this Excel file here. I'm not quite sure if I can open this right away here. Let me just see where yeah, this should be the one. So Butanediol basic and GC-opt summary, that was just now. Great. So sorry if my laptop is a bit slow here. Right, so this is this looks kind of handy. There's only 3 design solutions. Here you see, make this the objective value that the particular solution had within the MLP problem. You see here the number of interventions, and it says actually 4. We allowed 5, but you can also see here interventions excluded, that for every design at least 1 intervention. Here heterologous insertion, here some deletions are actually thrown out because they're not significant for the growth coupling. And this is one of the most important columns. These are the interventions. These are the reaction IDs that you can also find in the model itself. If there's no HR in the back of the reactions, it means that this particular reaction is deleted. Yeah, so in fact here we don't have any heterologous reactions. So we have 3 designs, each of which have 4 deletions. Here are the corresponding genetic interventions. So these are basically based on the gene-protein reaction relations within the model, which I can quickly show you. Let me just let me just do it like this. So we have the model. If we go to any kind of reaction, so for example, pyruvate kinase. So if you go to tab, you can. This is also handy, but yes, it works actually here. You can auto complete also the reaction that this model has, and then you have for this reaction some genes that catalyze or that code for this particular reaction. So in this term here is it's 2 genes. So this B1676 and B1845 that codes for this gene. And these genes, if this, for example, would be a knockout, these genes are taken and then put into this column here. So you can relate the reaction interventions to actual genetic interventions. Right. So another interesting thing is the maximum growth rate of the strain that is basically left after these interventions. And you also have, and this is a kind of a measure for the growth coupling strength. Sorry, this is the target flux, meaning in our case, the Butanediol production rate at maximum growth. And this is actually the minimum target flux. So you can nicely see here that at maximum growth, we have a positive Butanediol production rate, meaning we have a growth coupled system. And if you want to know how strong the coupling is, we can actually analyze a particular solution with a couple of more methods that are implemented. And again, we can use the Flux-based projection method here that I introduced earlier. And here you can specify a particular design that should be analyzed. So here I already put the design 3, so you can have a look here at the ID of this particular design. So this is design _3. Yep. Yes, I put this here. So this particular design is loaded and flux projections are plotted. Right. And as you can see nicely here, you have a fairly good growth coupling. As you can see, the lower flux bound here is well above 0 and actually particularly high for the maximum growth rate of roughly 0.15. So this, in terms of the flux space, looks like a good solution. Here you have the yield space, which looks pretty much similar to the flux space. And what you can also do here, we use the model that we have in the strain design analyzer, which has all the design loaded, so the 4 reaction knockouts loaded, and we create again a flux distribution using this FBA or parsimonious FBA, and then I plotted this again to an Escher map. Let me do a full screen mode. Right. So as you can see here, for example, glucose is still taken up. That's good. Also via the PTS system. And for example, you see one interesting thing here is when we look at the design 3, wait a minute. There is, I looked at the wrong column. So we actually, you have to look for this ID here. That's a bit confusing. Have to change this. You have to look at this column here. So this is design 3, the lower one. And you actually see here that the PGI, so the glucose 6-phosphate isomerase, is deleted in this particular design. And you can nicely see here that no flux is going through PGI, so sanity check successful. And we actually have a flux through the through the pentose phosphate pathway, quite a significant one, obviously. So yes, and in addition to that, so this is a flux distribution at maximum growth, and we can see here this is the merged Butanediol reaction. We can see here we have quite a significant flux of Butanediol exchange or synthesis at maximum growth. So this implicates a quite high growth coupling. And now, of course, you could really go into the details and analyze how does the flux go through this perturbed network. So the first thing we already have, so it uses pentose phosphate pathway. You can analyze things, for example, like the redox cofactor. So you see that a lot of NADPH is produced here. You can actually see here that the NAD trans hydrogenase is quite active. So just look at the right arrows here, right? So a lot of NADPH is transformed, I'd say, to NADH. Well, reason could be is, of course, that the Butanediol synthesis pathway takes a lot of NADH. Another interesting thing here is, for example, that still we have quite a significant lactate exchange or a production of lactate. So we could, and you could well try this on your own. So we could presume, for example, a knockout of the for example, the lactase dehydrogenase to avoid lactate production, and see what the algorithm proposes then for a growth coupled system. If we disable lactate production, we also have an acetate, quite a significant acetate production rate. Also because one of the steps within the Butanediol synthesis pathway involves the production actually of acetate itself. So this is not too much a coincidence. Here, of course, it looks a bit weird also to my eyes now, because we have somehow there's a fumarate coming out of nowhere in terms of this map. But as Dan was showing you, you could actually, well, maybe we can simply try this. We could actually have a look here. Wait, this is actually kind of good, because we already see the flux rate or the flux rate of other reactions that lead to this node, to this fumarate node, and we can see what are the significant fluxes here, which is actually this one. Let's see. All right, so we can see here that actually the fumarate is coming from L-aspartate. Produces ammonia. Yeah, so this is now really up to you how to analyze these design and these flux space that you generated with these designs, and draw a conclusion. Does this design make sense? Do we need to set other constraints to the model to make it more sensible, or to arrive at the conclusion that yes, this design, this growth coupling strategy is actually one that we for example should try in experiment. And I believe a design, build, test, and learn cycle you will always have in any probably metabolic engineering projects, also when using these kind of models. So yeah, just make sure you analyze these fluxes appropriately. All right. Okay, so this is the end of the basic examples. So what we basically did, the major points are setting up your model, getting your configurations file and setting up some parameters. So you find your results. So you specify how many number of interventions you want. Time limitation processes, this depends on your machine, of course. And yeah, hopefully with the help of at least this script, you're already able to run growth coupling algorithms and calculate solutions for growth coupling designs on your own. And we do this in the advanced examples. Now you can actually play around with the number of interventions. You can play around with different fixed growth rates. So you will also find a different solution based on the fixed growth rate that you choose. Because, for example, if you set a very high fixed growth rate, let me just switch to the example here. So this is the wild type flux space for our Butanediol producer. If you set a very high fixed growth rate, let's say here, at the same time means that the model or the designs has to keep or retain a growth of at least this fixed growth rate. So if you have your fixed growth rate somewhere here, it means it can perturb your model quite heavily and reduce the growth at the same time quite heavily, and you still have a feasible solution. If you have your fixed growth rate, let's say somewhere here, your range of possibilities for deletions at most are quite limited. But it depends on what you want. Yeah, I recommend to try multiple parameter sets, and that's actually what's done in the more advanced example, which I can quickly go over it, so there won't be too many new things and surprises here. So I would like to give you some time, hover over this the script, the basic script, maybe also have a look at the advanced script already, and yeah, give you the time to ask questions, anything.
So Tobias, I have a question and a suggestion. So I mean, this would be good to point out what is different in the advanced workflow compared to the basic workflow.
Okay.
I don't know if we have time for everything, but I was curious to your point about trying different max growth rates and things like that. Obviously, there's a massive number of simulations, and the simulation time builds up. But there's one decision in particular I was curious about, which is the microaerobic condition. And I understand that cells do experience oxygen starvation in large culture, but would it be possible that allowing more oxygen could break the growth coupling? Like if you like, how many of these designs would work in a more aerobic condition? Is that another thing that should be swept?
That could possibly be the case, and let's try it out. Right, because we actually, let's start here. We have our model and the design loaded here, and we can actually access let's do it here, the model here. Let me just see. Yeah, the design is loaded here. When we simply do STA and the model is within that that module, and then we can just use it as we would do with a simple CobraPy example. So reactions X, does it actually it actually does it, great. So we get to the oxygen exchange reaction, and we set the lower bound to, so we increase the possible oxygen uptake rate to, I don't know, let's set it to 10. So memorize these flux plots, and let's see what happens if we run this again. Yes, and this is actually what happens. So your growth coupling actually diminishes quite significantly. So obviously this design is really focused on a microaerobic, anaerobic environment here. I did this actually because I'm not so sure right now, but I think some of the enzymes need or are susceptible for oxygen. It doesn't work quite good in an aerobic condition. At least also in the paper, in the original paper by Yim, they do all the experimentations, also some modeling stuff in anaerobic or microaerobic conditions.
No, it's really, I mean, I don't think it's a problem or anything. It's just interesting when you start to consider how many variables you have to play around with and how many alternative designs there are out there in metabolism, especially when you consider heterologous reactions too. It's really just a mind-boggling number of things to explore.
True, and this is definitely a variable that you can also play around with. So you could go and when setting up the model, just unconstrain the oxygen uptake and see what happens, what kind of designs you get in comparison to an anaerobic or microaerobic environment, and see if you find similarities or something completely different. Obviously here you probably will find drastically different strain designs, if at all, in aerobic conditions. That's of course the question also. But yeah, that's then up to the user to make sense of this kind of algorithm and the models.
Just another general question. Sorry, feel free if anyone else has questions, just unmute yourself or raise your hand or anything. But I really like the principle of maximizing the minimum flux, because it's just hard to get a nice production envelope otherwise. Are there any weaknesses of this growth coupling algorithm in general, or are there any ways that, like, OPNOC or prior methods, are there any things that they do better, or is this just kind of unilaterally better at this point?
Yeah, in terms of growth coupling, I would say this algorithm is kind of straightforward, because as you said, it literally bases on the notion of these flux spaces and increasing of the lower flux bounds. But it depends how you define also growth coupling, because I mechanically looked at this lower flux bound. If you somehow define it differently, you will also find other approaches to the same problem. I mean, there's, for example, like the elementary mode analysis, which simply takes a different approach, but also have a very intuitive, let's say, representation of your model and your reconstruction, and build on that and use that to make predictions for growth coupling designs, just to take out modes that do not couple production with biomass, for example. So this is also very intuitive. It's basically same but different. I think, in my opinion, for a constraint-based approach, this growth coupling principle is quite strong. And if you're not fixed to one particular simulation, but you scan some parameters and evaluate different results, I think you get a pretty good idea of what growth coupling strain design could work. I mean, the premise is always the model itself, and I think we figured that out also with our current projects, that even though the models have been highly curated, maybe some information are still missing, some links of enzymes that maybe can catalyze reactions that we haven't dreamed of before are actually happening, and overtaking some functions, which then crushes our growth coupling. This can always happen, and this is a bit hard to predict by now, and where we still are in need of, well, this design, build, test, learn cycles. So yeah, so this underground metabolism and promiscuous reactions, promiscuous enzymes, this is one of the biggest issue, I think, for this kind of growth coupling.
That makes sense. Any other questions or comments from people? If people have any comments on the short course organization in general, or especially things that you wish we had covered, feel free to email me or Tobias afterwards, because we can add this material into the repository. That's part of the point of having the GitHub, is, you know, it's not just a static thing. We can always update it. Maybe with the time that's left, if there's no other questions, maybe you can just give us a quick tour of the advanced notebook,
yes,
just to point out what is different.
Yes, that's a good difference, and it's actually the more advanced stuff focuses on, yes, exactly on this scan of parameters. So it actually does the same modules and same stuff is loaded. I actually skipped the model design part a bit because I saved this one earlier. You see simulation properties of the base strain is similar. The thing is, what I use here, the growth coupling algorithm, or this the growth coupling suite, is able to run multiple growth coupling experiments or simulations in parallel. So that means it can dedicate a certain number of processes to separate simulations where you can test different varieties of parameters. You would think, okay, maybe this doesn't make sense, because if I run one simulation, I can use all my processes that I have of my machine, and I will run this for some time, and then just simply run the next simulations. I will efficiently use all the capacities I have. Actually, what I find out with the MILP problem and Gurobi is that after a while, even though the solver couldn't find, the Gurobi solver couldn't find, or hasn't found the optimal solution yet, it somehow slows down. So it stops using all of your processes and your capacities. So in order to compensate for that, I started to do multiple separate simulations in parallel. So that's what you can do here. So you can set up actually a list of dictionaries here, where you specify different parameters which you would normally define in your config file. So for example, you can say number of total interventions. You have 3 separate simulations that you want to run in parallel, and you say, for each of the simulations, I want to have a different number of total number of interventions. And what you can also do, you can run several simulations in parallel, but you can also run simulations now in sequence and change the parameters. So this stuff is run in parallel and in sequence. These kind of parameters are also run with different fixed growth rates, and that's what I just told. Maybe it's a good idea to also investigate different fixed growth rates, and that's what here then in sequence. So to really get maximum out of your computer and your machine. So that's one thing. Have a look at it at the example. It's not much of a difference. It's the only thing that you have to take care of is this line, integrate it. Google for it why this is necessary. It's a bit complicated. Just do it. It's necessary to run multiple processes, multiple simulations. you can actually specify how many of these are run in parallel with the max workers here, and anything else apart from these two cells is basically the same. So at the end of the day, you will have, maybe I actually have, sorry, how do I come back? Already, right. So this is example that I run earlier. You will have multiple results file to the same problem, but with different parameters. I marked this here with the abbreviations, and the analysis routine then actually takes all the designs and all the results file into account and creates this summary file. And maybe, yeah, we have 5 minutes left. Let me just quickly, oops, that's not going anywhere. Oh yeah. Okay. That's not the one. Yeah, okay. Close this one first because it has the same name. And just that you believe me that we actually have the same. So you see that we have quite a significant number of designs. So 70 with different complexities, a different number of genetic interventions. There's actually one with 2 reaction knockouts. So you have a wide variety, and yeah, then it's up to a manual curation which reaction you like. Because what we also did here is, and this also belongs to the advanced example, is we actually now allow also to change the carbon source. So we allowed here to change one of the carbon sources. So the model automatically deletes, for example, the one that is predetermined, for example, the glucose uptake rate, and changes it with one of the source target that we have specified here. So I made a, so you can simply use this list, or you can only use, I don't know, glucose and an uptake of lactate, whatever, and the solver will automatically find the optimal solution or combination of deletions, add-ins, and carbon sources that maximizes growth coupling. Another thing is here that we also now allow for co-feeds. So in addition to our main carbon source, which we defined here, we also allow for the uptake of another carbon source. So a co-feed, which I specify. Okay, I didn't specify particular targets here, so that's so the choice for a co-feed is based on some other logics on the number of carbons. So I think there's, don't want to go into detail here. Number exchange carbon threshold, yeah. So so that only metabolites can be taken up that has a maximum carbon number of 8, for example. What the algorithm can also do, and this is probably the last major thing that differs from the basic example, you can actually evaluate the gene protein reaction relations on the fly when the solver gets a solution. And I've been showing you this when the solution occurred. Let me get to this example where the algorithm was running here. So for example, it finds a solution, a new solution, and then based on the original model, it evaluates if this kind of reaction, these kind of reaction deletions can actually happen, or can be made real by appropriate gene deletions. So it gets the reactions that are deleted for this particular design. It gets the corresponding genes, deletes the genes, and evaluates if other reactions are also deleted. and at the same time evaluates if you still have a growth coupling with the gene, rather the reaction deletions. And so you get a clearer idea right from the beginning, right at simulating these kind of solutions. You get an idea if this kind of solution will be feasible for you to implement in an actual cell, in an experiment. So this can be quite useful. Right. So right, this is done by setting this parameter to true. Yes, exactly. Just as a hint, you can of course, and I think that I did it here. You can, but you can simply ask me later if you want to know more about this. You can also specify essential reactions that you know are essential, but maybe the model doesn't really think it's essential. Just an example is, for example, enolase. You can perfectly knock out enolase in the model, at least for E. coli, and it will somehow happily grow. But in fact, the single gene deletion of the enolase is actually enough for to kill the E. coli cell, because it has another non-metabolic functions, I think, in the RNA degradation, something like this, which is essential to E. coli. Yeah, and with these, you have a quite comprehensive method at hand to get quite plausible idea about growth coupling designs using these M or metabolic models. Yeah. So if you have any question about this advanced type of algorithm, let me know. It's not much difference. It's just more information, more constraints, and a bit more realistic. Right.
Tobias, this is just fantastic. Thanks so much for putting this together. I kind of wish we had. I'm thinking we should have like a competition in the lab to see who can use the short course materials to do, a real research thing or something like that. Maybe you can try to encourage some of the younger students to try this out. Because I mean, you made it accessible for anyone to do realistic designs. Like it looks so easy.
Yeah, I mean, if you if you pass this point, basically, if you can load the growth coupling suit, then it's almost done, actually. So this is sometimes the critical point of getting the whole stuff installed, particularly custom modules. So I hope it's easily accessible. Yeah, it's really up to preparing the model, know what the model does. So yeah, to exactly get to know what you've been talking about before and presenting quite extensively, and then of course combine your biochemical microbial knowledge with the designs and the results that you get, and put it together and make sure that it somehow makes sense. So yes. So I would also recommend play around maybe with small models. If you really want to predictions, use the most current big models, get into a high performance cluster, and yeah, let it run.
Sure. Yeah, it's kind of limited by computing power at some point.
Yes,
okay.
But I found out, yeah, at least for the HPC, the high computing cluster at DTU, it works actually perfectly fine. So this can be done quite easily, surprisingly.
All right, so I think we should probably end on that note. Thanks for everyone for sticking around, and 4-hour session is pretty brutal, but I hope you got something out of it. There will be 3 more. We're doing independent component analysis of RNA-seq data, which has been a big success of our lab recently, next week, so that should be exciting. And then we have sequence modeling and kinetic modeling on the horizon as well. So very different topics. And yeah, we'll try to live up to Tobias's standard for putting together polished material, but I don't know, it's a high bar. We'll see how we can do. anyways, I'll be sharing the recording with everyone. If you want to review anything that you missed or send it to anyone who may have missed that you collaborate with, we'll try to get everyone access. It's just going to be figuring out a hosting solution. So I'll work on that. With that, thanks to Tobias for helping and putting together such a nice overview of his tools. And
it was a pleasure.
Thank you all for attending. All right, take care, everyone.